All right. I see some people still trickling in, but I think we can get started so that we can keep on time. Welcome, everyone. Good afternoon. This is a social behavior session. I'm Noah Pinter-Wollman. I'll be the moderator for the first half, and Julia Zoltz here will be monitoring the questions during the first half. And then at the break, we'll switch roles just to mix it up and confuse people, and Julia will be the moderator, and I'll monitor the questions. I'm really excited to be here. I've been trying to get to this meeting forever and ever, and every time something came up, so the virtual world actually made it happen. So I'm excited to be here, and I've been really enjoying all the talks and looking forward to this session. So the way we'll do it is I'll introduce the speaker. The speaker will go for the 15 to 20 minutes, and if there is time for questions, we ask that you drop your questions either in the chat or in the Slack channel for this for the social behavior. And Julia will find those questions and read them out. And so without further ado, let's get started so that we can keep on time. Our first speaker is Vincent Formica, who will be speaking about age-dependent mortality and reproductive improvement in a wild population of long-lived invertebrates.
Thanks, Noah. Does everything look good on your end? You can hear me?
Looks great.
Okay, thanks. So as Noah said, my name is Vince Formica. I'm going to be talking to you about about reproductive improvement and age-dependent mortality in a wild population of long-lived invertebrates. Before I get going, I want to acknowledge my co-authors, Vitor dos Anjos, who is my undergraduate student at Swarthmore College. And this is really a result of his ideas that pushed us into this world of demography, and Butch Brody from the University of Virginia. And before I go in too far, I'll say that this is our first foray into age-related demography. So we're looking forward to comments and suggestions as we go forward. So how we think about age-related performance and behavior is often kind of shaped by our own experiences and those around us. So this is how I am expecting my future to look. My dad likes to say that getting old sucks, and this is usually muttered when he stands up from the sofa. And really what he's getting at here is an idea called senescence. And we can define senescence as kind of a gradual decline in performance or fitness as individuals age. It's based largely on human and mammal data as well as bird data. And the metaphor you can kind of think of aside from humans is you can think of buying a car, right? As you buy this brand new shiny car and it slowly kind of craps out on you. And we know that in animals, this is due to somatic mutations and mechanical and physiological wear and tear that leads to like this deterioration of performance over time. My dad also has another idea about aging that he told me when I was a teenager, which was old age and experience will always beat youthful enthusiasm. And he used to say this when he thought I was up to something, and he usually caught me before I did it. So this idea is coming from something that we call age-related improvement, or if we're looking at reproduction, we call it reproductive improvement. The metaphor for this is, say, whiskey in a barrel that's aging, or wine, it's getting better with age. So we can define this as kind of an increase in performance or an increase in fitness, an improvement in fitness over time. So there's a lot of evolutionary models for why this might happen, and kind of the story or the narrative goes kind of like this. As you age, you're more likely to die. And so you start investing more in performance, reproduction. You worry less about things like predation risk or investing in maintenance. So this is mostly seen in fish and amphibians, although there are a few mammal models where we see this too. And both of these ideas are related to a concept called terminal investment theory, which is a subset of life history theory that just says that as individuals age, they should change where they're investing their resources, depending on their likelihood of mortality. There are very few studies in invertebrates. There's some really great work in age-related changes in crickets coming out of some Spanish labs, but there's very, very few work in long-lived invertebrates. And so we just set out to use a data set we were collecting to study social networks to look at a natural history question about aging in a long-lived insect. So our questions are just, do long-lived invertebrates experience reproductive senescence, or do they experience age-related improvement? Or is there just kind of another paradigm out there that we don't know because we haven't really studied long-lived insects? With our data set, we're also able to ask, are there sex differences in age-related reproductive changes? We know that the sexes can invest differently in reproductive strategies, so it seemed like a natural question. And then lastly, this terminal investment theory, which I see as kind of ruling age-related changes, at least in evolutionary biology. It relies heavily on this kind of patterns of mortality and survivorship. So we wanted to see if that matched the other results we were seeing in performance. We did this in our study system, the forked fungus beetle. They're a pretty cool insect. They're long-lived. There's been one individual that was tracked by another team that lived up to 8 years. Most of our beetles live 1 to 2 years, although we have a substantial portion of our metapopulation that's living to 3 and 4 years. Another neat thing, if you're interested in the evolution of morphology and weaponry, is that their size is determined when they eclose from pupa, and it doesn't change. It's static for their whole lives. They spend most of their breeding season on a single log, so they live on these shelf fungi of rotting logs, and they don't move very much. So about 12 to 20% of a single subpopulation might move to another subpopulation, but the vast majority are kind of there in one society over the whole breeding season. If they do move, they don't move very far. Their median movement distance is about 50 meters to another rotted log with a fungus on it. The males have these horns. You can see these thoracic horns here, and also these clapial horns that are a little harder to see that they use to fight over access to females. Females don't have those horns. And these horns are under really strong sexual selection. Forked fungus beetles are kind of a classic sexual selection system. Females are also under strong selection for body size, even though they don't have horns. Males prefer larger females, and larger females lay far more eggs than smaller females. Forked fungus beetles also have these really informative behaviors that we can just observe with the naked eye that indicate reproductive effort and reproductive success. So part of their kind of mating rituals, it starts off with a courtship, which we're going to call reproductive effort. And so this is a male who's climbed onto the back of a female, and he faces head to abdomen, and he does this little dance. He shakes back and forth, and there's a whole bunch of other behaviors that go along with this courtship. Females have complete control over reproduction. They've got an anal sternite that protects their genitalia. So if they accept his courtship and his copulation, he'll transfer a spermatophore, a giant spermatophore, to the female. And only if he has successfully transferred that spermatophore, he'll then turn around and face head to head and do this mate guarding behavior. Both of these behaviors last for several hours. We've documented males courting females for over 20 hours in the lab, and we've used time-lapse photography to see in the wild that males will guard for upwards of 12 hours after successful insemination. So we're going to call this reproductive success for both the male and the female. Females, we have another component of reproductive success, which is egg laying. And so females lay a single egg on these fungal brackets, and then they glue it down, and they have this kind of like, we call it like a butt waggling behavior, where they're just like gluing the bracket down and putting this substance that hardens over the egg onto the bracket. I'll show you a real quick video here of both of these behaviors. So this female in the background, you can see she's laying an egg, and she's doing this kind of butt waggling behavior. These are eggs that are glued to the surface. This female is also laying an egg here, but while she's doing that, a male is trying to court her. So he's dancing on her back and banging on her carapace with his legs and his antennae. Yeah. Okay, so how are we looking at age-related changes in individuals? So we observed 19 what we call focal subpopulations. So these are logs infected with fungus. And our focal populations are the largest subpopulations in our metapopulation. We've got about a metapopulation is about 2 square kilometers at Mountain Lake Biological Station in Virginia. And so the focal populations are the large ones in the forest. And we studied all of these populations from 2015 to 2019. We captured all B. cornutus, we photographed them, we labeled them, and then we returned them within 48 hours. And we do this every year. All focal subpopulations are scan sampled 3 times a day for 60 days during the breeding season. So we've got a team of undergraduates that go out with flashlights and mirrors, and they're looking in every nook and cranny on these logs to try to find all of the fungus beetles. All behaviors and locations of all individuals are recorded every scan sample. Okay, so how do we age beetles? I wish I could tell you that they were like me and as they get older, they're going from a dark phenotype to kind of a lighter gray phenotype, but that's not how it works. We have to use mark recapture methods. There's no way to kind of age them morphologically. So we labeled all the beetles that we could find in the meta population, in our focal populations and these peripheral populations around them. And then we had to throw out all that data since we didn't know the age of anybody in 2015 in our first year. That first year and subsequent years, all fungal patches within 100 meters of these focal populations, so 2 times the median movement distance, are also searched 3 times a summer for newly enclosed beetles. Preliminary data tells us like 3 visits will get you nearly all the beetles, like 85% of the beetles. Any unlabeled beetle in our subpopulation that we found in subsequent years, we could consider them to be at least 1 year old. So we're doing like a 1+ kind of aging system here. So we think we're getting pretty close to finding and labeling most of our new beetles in the meta population. If we're finding a beetle and we find it unlabeled and we assume it's young, that should kind of not give us a signal. So we think that this method is pretty good and we're being pretty conservative in how we're aging our beetles. Okay, when you do this kind of work, there's 2 ways that you can do demography work, either cross-sectional or longitudinal. So cross-sectional is like this. You go out, say, in your 4th year, you've got everybody marked, you know the ages of all the individuals. I've got these here as dark, as young and moving towards gray and white as they get older, just as a visual cue. So you go out in year 4, you've got a subsection, you've got a subset of your population or your whole population you're monitoring, and you're basically comparing older beetles to younger beetles and you're comparing their performance. So this is nice because you get a large sample size and you can do kind of sophisticated statistical modeling. You've got some more good statistical power. The problem is that you could have a filtering effect, right? So these older beetles, they might be around, they might live to be old just because they're awesome. Like they've been awesome when they were young and they're still awesome and they've survived. And so you're kind of comparing everybody to just the Judi Denches of the world that just, have always just been awesome. So what we do in order to add to these cross-sectional data is we do what's called a longitudinal data. So a longitudinal analysis. So this is where you track cohorts over time and you're comparing the older individuals, not to current younger individuals, but to their younger selves. And so you're tracking cohorts over time. This is really labor intensive. You can't miss a year and you have lower sample sizes just because not everybody lives to a ripe old age. So we did both of these. So here are the results from our cross-sectional model. So this is for inseminations, and we're looking, these are generalized linear mixed models. And so we're looking for differences in age, but we're also interested in both sex and really this age by sex interaction. So a cross-sectional analysis, so we've got about 1,500 individuals. This is data just from our focal populations. And so what we're seeing is kind of classic age-related improvement or reproductive improvement. As beetles get older, they just get better. So they're having more inseminations, both males and females as they get older. So when we, but there's no differences between the sexes in this pattern that we're seeing. So when we move to our longitudinal model, right, the statistical power is a lot lower, so we can't have as sophisticated models. So we're just looking at age, sex, and body size here. And what we're seeing is kind of support for our cross-sectional data that both males and females are getting better as they age. These are still significant, even though we've got a lot fewer individuals, only 96 individuals across these 3 years. Okay, so that's kind of reproductive success. So let's look at investment in terms of courtships. So what we're seeing here is still age-related reproductive improvement, but we're just seeing it in males. So older males are able to get more courtship attempts. They're putting out more effort as they get older, but females are not. The female line is not significant. So there's a lot of biological reasons for why we might see this. One of them is that we think that courtships are mostly initiated by males. They're the ones that climb onto the backs of females, and we haven't observed any signals from females kind of soliciting those courtships. So that's one possible explanation. So if we take a look at the longitudinal data here, we don't have the power to kind of test the differences in those slopes. So we're seeing support for it, but we're not seeing those differences between the two, but that's how we set up the model. But still, it's supporting our cross-sectional data. So we're kind of happy with how the gold standard, these longitudinal models kind of support the cross-sectional data. Okay, so for females, we've got one more measure of reproductive success, which is egg laying. And we're also seeing the same pattern that older females are just laying more eggs. Now, we don't know the quality of these legs. We just know that they're observed laying eggs more often. And so the cross-sectional and longitudinal data for this are identical. The females are just getting better with age as well. Okay, so after we saw these data, we were wondering, reading more about terminal investment theory, we were wondering if there's kind of age-dependent mortality in forked fungus beetles. And so we did a Bayesian mark recapture analysis using this program called BASTA, which I'll highly recommend. It's really, really easy to use and gives you some really great figures to kind of visualize what you're doing. I won't go into the details, but if you're interested, this is the models we're running. And so what we see is, yeah, there is age-dependent mortality. So as beetles get older, they're more likely to die, more than you might expect just by kind of probability, just by moving along. So what I mean by that is you got this kind of upward curve, right? So as you get older, you're more likely to die. And this is significantly different between males and females. I don't want to make too much out of that because you don't start to see it until much, much later in the projected lifespan. But we're kind of seeing a type 2, maybe a type 1 survivorship curve here. I should point out that we don't have the early life between the eggs to pupa. This is just from a closed adults here. But we are seeing age-dependent mortality. So in conclusion, from these data is that male and female forked fungus beetles, they're engaging in more successful inseminations as they get older. Males are putting in more effort with courtship and data I didn't get to talk about here is that we've done another study that we're showing that males are getting more aggressive as they age, actually. So this is more evidence that they're kind of investing more in reproduction. We think that this is pretty good evidence for reproductive improvement, not senescence. And our age-dependent mortality suggests that terminal investment theory is at work here and is a good predictor of why they might be doing this. So a beetle's model might be go all in before you die. So why are Butch and I doing this, right? Like we're not demographers by training, as I said in the beginning. We study social behavior. And so one of the reasons that we're thinking about this is that we've noticed in, like a lot of other folks, that we're seeing pretty high variance in selection, the magnitude and direction of selection in traits that we measure, especially social behavior traits. So this is an image out of our recent Journal of Animal Ecology study where we looked at social network traits, and we found that there's really highly variable there are differences among the subpopulations in magnitude and direction of selection for these social network traits. So we're wondering if maybe age structure of population is a kind of mostly ignored demographic parameter that might help us explain some of the variance we're seeing in sexual selection and multi-level selection. So with that, I'll thank my collaborators. And I'd also like to thank Debbie Roach, who kind of pointed us at looking at longitudinal data as well as cross-sectional data, and Fernando Colchero, who really took an immense amount of time to help us to get the BASTA files, the BASTA analysis working, and spent a lot of time over Zoom with us. So I'd like to thank him, as well as all the undergraduate army that kind of helps us collect that data. So I'll leave you with a thought that getting older doesn't have to suck, at least as long as you're a forked fungus beetle. And with that, I'll take any questions if there's time.
Awesome. Thanks, Vince and Vince's dad, for that excellent presentation. We have 2 questions so far. So first question, Giuliano Morimoto is asking, did you check whether or not old males inseminate more young versus old females? Also, did you check any trait of offspring? And could these effects have showed a trade-off by Lansing effects? I'm actually not sure what those are.
That's okay. I'm not sure about that last part anyway, but those are great questions. So the first question was like, do males mating with older or younger females yet? No, we haven't looked at that. I'm excited to see if males have a preference in age of females. It takes a really long time. It takes 3 to 4 years to raise those beetles. So we're in the process of doing that. Trade-offs in the young, that's something Vitor wants to really look at, and seeing if the eggs that females are laying might be lower or higher quality, or if the larvae of old females or old males are different. And we haven't looked at that, but it's on the docket.
Awesome. They then clarified that Lansing effects refer to offspring from old parents are worse off. So,
oh, cool. Great. We're definitely going to look at that. And now I know what to call it. So thanks.
Awesome. So Ambika's asking, when during guarding do females lay the eggs?
It's not clear. It's not clear when that happens. It might happen at the end of guarding, but we think that it may take much longer than that. So it's interesting as females can store sperm for over a year. And so it doesn't necessarily happen during the guarding event. So yeah, that's the answer to that.
Okay, I think we have to move on, but great job.
Right.
Thanks so much.
Thanks, Vince. Thanks, Julie, for taking care of the questions. All right, our next speaker is Sahas Barve, who will be talking about cooperative polygamy, is a reproductive alternative, not a reproductive compromise in a social bird.
You're muted, Sahas.
All right, let's try this one more time. Screen. I guess you're muted is the most commonly said thing in 2020, continuing on in 2021. So hope you can hear me now and see my screen. So I'm Sahas Barve. I'm a postdoc at the Smithsonian National Museum of Natural History. I'm an avian evolutionary ecologist with interest in life history evolution, and I have field sites in the Indian Himalayas and Carmel Valley, California. And today I'm going to talk to you about lifetime reproductive fitness benefits of a cooperative breeding behavior. So cooperative breeding can be loosely defined as a reproductive strategy where more than two parents help raise the offspring. And in sexually reproducing animals, there are some adults amongst the parents who are helping raise non-descendant kin, or in some cases even non-kin. And of course, raising non-descendant kin is much worse from a fitness point of view than raising your own offspring. But kin selection theory suggests that these helpers who are helping raise non-descendant kin accrue inclusive fitness benefits by helping raise closely related individuals. There are two main forms of cooperative breeding, roughly speaking. In systems where there are non-breeding helpers, these are often offspring from previous reproductive cycles that stay back in delayed dispersal and reproduction to and then help raise subsequent progeny in the group. And then there are cooperative co-breeders, which are like these lions, form cooperative co-breeding coalitions that stick together all their life and communally bring up offspring at every reproductive attempt. Generally speaking, because in both these cases, adults end up helping raise non-descendant kin or not direct descendants, these strategies are considered a best of a bad job strategy that adults use when they can't breed independently. But there is some evidence in birds like red cockaded woodpeckers, green wood hoopoes, and even red wolves that suggests that cooperative breeding might be actually an alternative life history strategy that gives equivalent or even higher direct reproductive fitness benefits than dispersing as an adult as soon as you can. So in these systems, helpers gain survival and fitness benefits from staying as helpers for a few years. And these helpers then end up having higher lifetime direct fitness compared to individuals that do not help. Cooperative polygamy is sort of an evolutionary enigma because unlike systems where, for example, cooperative polygamy here can be defined as any system in which the breeding group consists of polyandrous, polygynous, or polygynandrous groups. Cooperative polygamy is problematic sort of because co-breeders compete for fitness at each breeding attempt. And because these co-breeding coalitions are lifelong, they sort of present a lifelong reproductive compromise for each one of these individuals as they try to maximize their own individual fitness at each breeding attempt. And so, rightly so, short-term studies of cooperative polygamy have shown that this is a best of a bad job strategy. And individuals only resort to cooperative polygamy in the absence of independent breeding opportunities. But long-term studies of cooperative polygamy are truly what are required to understand whether the lifetime fitness benefits of this strategy sort of lend it to be a best of a bad job strategy versus this alternative reproductive tactic, which gives you equal or higher fitness benefits compared to single breeding or independent breeding. So we asked just that question in a long-term study population of acorn woodpeckers. This population has been studied for the almost 50 years continuously at Hastings Reserve in Carmel Valley, California, which is Hastings is one of the oldest biological research stations in the country, and actually only a 45-minute drive from Asilomar, where this conference is usually held. Acorn woodpeckers are famous for building these large acorn granaries, which are individual holes drilled into the bark or dead parts of the tree. And these granaries define acorn woodpecker territories. They can have several hundred to tens of thousands of holes that accrue over generations of acorn woodpeckers. And acorn woodpecker survival probability is significantly higher in territories which have granaries. And especially males benefit significantly from having a territory with a large granary. So within the 50-yard groups that we study in our continuously monitored population, we have groups that breed as a simple pair. There are groups that have a polyandrous sex ratio. Some groups have polygynous sex ratios, and even groups that are polygynandrous, where multiple males and multiple females communally and cooperatively raise a clutch of offspring together. And each of these types of breeding units can may or may not have non-breeding helpers helping them. And these non-breeding helpers may be both male and female. So the frequency of cooperative polygamy varies by sex. In males, cooperative polygamy is much more common and is about 50-50. So about 50% of the groups have multiple breeder males in the group, while the other half have a single breeding male in the group. And within those cooperative breeding males, most of those are co-breeding duos, and this will be important later in the talk. Within females, cooperative polygamy is much less common, and about 80% of the groups in our population in any given year have only a single breeding female. And again, within cooperative breeding females, duos and trios are common. So we wanted to ask, what are the short-term and long-term direct benefits of cooperative polygamy? And this data comes from parentage assignments of over 3,300 nestlings that we made to their biological mom and dad, so we have data for lifetime direct fitness for about 300 males and 300 females, but we restricted our analysis to about 256 males and 236 females, individuals that were only that were born up till 2006. So we basically wanted to control for any bias of lifetime reproductive success of short-lived individuals. So we took those out and let each woodpecker be alive for at least 10 years, which is about twice the normal life expectancy. So cooperative polygamy is in fact a short-term compromise for both males and females. So in any single reproductive attempt, a single breeding male or single breeding female has significantly higher direct offspring than cooperatively co-breeding individuals of either males or females. But for females, co-breeding is a compromise throughout their life. So these cooperatively joint nesting, as we call them, females, do not have an advantage in the age at which they first start breeding. These cooperative females do not have longer reproductive lifespans, or do not have more nesting attempts, which results in single nesting females having significantly higher lifetime direct fitness than joint nesting or cooperatively co-breeding females. For males, however, co-breeding is an alternative reproductive strategy. So for males, they don't incur benefits for the age at which they start breeding, but co-breeding males, especially duos, see an increase in their reproductive lifespan and the number of nesting attempts they have throughout their life, which results in reproductive. So co-breeding duos having the highest lifetime direct fitness of any other type of the category. And when you lump these co-breeding males together, in general, cooperatively co-breeding males have a lifetime direct fitness that is not significantly different from a single breeding male. So whether they breed cooperatively or as single males, they have about the same number of direct offspring throughout their lives. So interestingly, co-breeding is both a best of a bad job and an alternate breeding strategy in our population of acorn woodpeckers. We found that within the same population, for females, co-breeding is a best of a bad job strategy, and for males, co-breeding is potentially an alternative breeding strategy. So why do females cooperate, and why do males form large coalitions in the first place? I think it all goes back to the fact that these acorn woodpeckers are really, really closely tied to these acorn storage granaries. And the granaries define an acorn woodpecker territory, and the number of granaries in the landscape is limited, and hence the number of the dispersal opportunities for individual acorn woodpeckers are also limited. And so what we see is that in both males and females, the incidence of cooperatively co-breeding with same-sex individuals increases with population density. So as population density increases, the number of breeders of any one sex, both male or female, in any given group also increases. So they become more cooperative as independent breeding opportunities go down in the population. But males also get a benefit from co-breeding in acquiring territories. So we see that while females can't really acquire large or better quality territories when they are co-breeding, a large coalition of males is more successful at acquiring a high quality territory. And so one tactic they might do is 3 or 4 individuals might form a coalition and fight together at a breeding vacancy to win a high quality territory on which they can live for a long time and have a lot of babies. So long-term studies, and this is condensing almost 50 years of lifetime fitness data, have shown us that while at the short term, co-breeding may be a cooperative compromise because cooperating individuals bear the fitness cost of competing with their same-sex partners at each breeding attempt. Long-term studies like ours can show that that cooperative co-breeding can actually have increased lifetime fitness benefits for some individuals in the population compared to single breeders, and that the benefits of cooperation may vary by sex within the same population. With that, I would like to thank NSF and my lab at Old Dominion University, and the Hastings Natural History Reservation, and the over 200 people who have worked on this project over the years. And I will take any questions. Thank you.
Okay, we have lots of time for questions. Oh, here's one. What is known about the mechanisms affecting whether any particular male or female participates in cooperative breeding? So who's most likely to be a cooperative breeder?
That's a great question. And so it sort of depends on, like I said, about on the availability of territories. And so if especially if there are only good quality territories available that year, you need a big coalition to go fight for those good quality territories and win those territories. So dispersing itself may be dependent on whether you are ready to cooperate with a closely related same-sex individual or not.
Okay, so Noah's interested in the elephant in the room, which is inclusive fitness. So
Sure.
what is known about inclusive fitness in the system? Are these guys related to each other? Okay, you're nodding. Go for it.
Yeah, so that's an obvious and great question. The acorn woodpecker population at Hastings is generally pretty highly related to each other. And so I think each individual is about 0.3 related to any other woodpecker in the population. And so there's obviously inclusive fitness benefits. It's a little difficult to model inclusive fitness benefits. And so we are sort of doing that right now. But I just wanted to show that you don't really need inclusive fitness benefits to show that that cooperative co-breeding can be an alternative reproductive strategy. You just have to use direct fitness benefit data to show that. And so that was sort of the focus. I mean, of course, so the flip side of that is that if these co-breeding duos have higher reproductive fitness than single breeding individuals, especially in males, inclusive fitness will only add to that. And so co-breeding duos may have significantly more fitness overall than single breeding individuals.
Right, but it might change your answer about the females.
Sorry?
It might change your conclusions about the females.
Yes, it will. Yeah.
I also think that would be really fascinating to look at within those groups that had more than two individuals of the same sex. So like maybe if the two males are brothers and the third one's not, and like that would change.
Yeah, absolutely. And actually, and that's one of the reasons why the inclusive fitness becomes a little more complicated, because they may cooperate with their dad or their brother or their first cousin or their second cousin based on who their nest mates are. So they often end up being coalitions, end up forming with birds who they think that's their brother or sister, but they might not directly be their brother or sister. They might be their first cousin. So,
whoa, interesting.
Yeah.
Maria Servideo is asking, is it known whether, well, very interesting talk. So I'm cutting out those
Sure, thank you.
exciting parts, but I encourage you to read them later. Anyway, is it known whether cooperative breeding affects survival?
Yes. So we know that group size affects survival. So birds that live in larger groups survive longer, and birds that there's potentially an effect of group size and territory quality. So groups that live on high quality territories, which are in our case are territories with big granaries, individuals survive longer. And that generally these effect sizes are bigger for males than for females. Females tend to get the short end of the stick often, and they don't survive as well as but they do survive better than helpers. So that's something.
Okay, I'm not seeing any other questions, so I'm going to ask my own quickly.
Sure.
I kind of lost track of where all those different data points came from, but is there a way to put together some of these fitness effects with the information about territory saturation? So like maybe cooperation is more favored when it's more competitive to get a good breeding territory or something like that?
Yeah, so we showed that last year for females, that the only sort of model that explains why females may cooperate is the habitat saturation model. So as independent breeding opportunities go down in the population, with population density going up, females tend to cooperate more. So the incidence of two sisters or a mom and a daughter co-breeding and joint nesting together increases with population density. But that's also seen for males. And so in general, habitat saturation probably lends to cooperative breeding, but there are other factors as well, because they rely a lot on the availability of acorns in the system also.
Okay, I think we're almost ready to move on, but I'm imagining a world you could produce where you give them unlimited granary opportunities and see if they evolve more or less cooperation. That would be really cool. Not sure if
we are doing that. We are doing a grad student in the lab is doing that right now. So we are adding and taking out acorns and leaving some with their granaries intact to see what happens.
Awesome.
Yeah.
Exciting. Looking forward to that. Okay.
Thank you so much.
Well, and I'm looking forward to all the inclusive fitness complexity and giving that as a question to my own behavior class.
Definitely. Thanks.
Cool. Thank you so much. I think the next talk, the speaker is here, but we also have it recorded. So I think we're probably going to show the recorded one, but then the speaker will be here for questions. For some reason, I can't see part of my screen, so I'll just read from the program. Our next speaker is Michael Bertram, and he'll be talking about sex on drugs and covering impacts of pharmaceutical pollution on complex behaviors and sexual selection processes in fish.
Okay, thanks very much for listening. I'll be talking about some work that my collaborators and I have been doing, investigating impacts of pharmaceutical pollution on complex behaviors and sexual selection processes in fish. So we know that pharmaceuticals are used across the globe for human and veterinary healthcare. But over the last 10 to 15 years, there's been a growing recognition that pollution resulting from the production and consumption of pharmaceuticals poses a major threat to wildlife, ecosystem functioning, and human health. Despite this, the vast majority of countries worldwide still have little to no regulation concerning the discharge of these drugs into the environment. To give you some idea of the pace of this problem, this graph shows time on the x-axis from 1955 to 2015 and proportional change since 1970 on the y-axis, with 0 or no change being indicated by the horizontal dotted line. So this curve represents the rise in carbon dioxide in the atmosphere over this period. Here we see the growth of the total human population. This curve shows the increase in the total output of the chemicals industry across all developed nations. And this rather vertical line shows the increase in global pharmaceutical consumption. And indeed, this solid line doesn't tell the whole story, considering that the x-axis here ends at 2015. Indeed, the rate of growth in global pharmaceutical consumption has only risen a further 24% since then. So where are we up to as of 2021? Well, the number of pharmaceutical doses dispensed per annum globally has now reached 4.5 trillion. There are currently around 5,000 different pharmaceutical substances on the market, and more than 600 of these have now been detected in ecosystems and organisms all around the world, spanning 71 countries and covering all continents. And these drugs possess a range of characteristics that make their prevalence in the environment particularly concerning. This includes, for example, the fact that pharmaceuticals are specifically designed to have biological effects and often at low doses. Various pharmaceuticals can be highly temporally persistent in the environment. Pharmaceuticals can act on physiological systems and pathways that can be highly evolutionarily conserved, even across diverse taxa. And there's even evidence that certain pharmaceuticals can not only bioaccumulate in the tissues of exposed wildlife, so be taken up into the tissues of organisms, they can also biomagnify in food chains. And what this means is we often see higher concentrations of pharmaceutical drugs in the tissues of animals as we move up trophic levels, and often see the highest concentrations in apex predators, for example. And although pathways of pharmaceuticals into the environment are highly diverse, one of the major routes is runoff of veterinary pharmaceuticals that are used in agriculture. Now, use of veterinary pharmaceuticals in agriculture is actually primarily for the prevention and treatment of disease. However, vast amounts of hormonal growth promotants, or HGPs, are also administered to cattle worldwide to increase their rate and extent of growth. Hormonal growth promotants are most often administered as slow-release subcutaneous ear implants. They typically contain a mixture of natural and synthetic hormones. So these are 3 leading brands of hormonal growth promoting implants: Revalor, Sinovex, and Compudose. each of which have different formulations, but what you can see is they share in common high doses of the primary ingredient, trenbolone acetate. So trenbolone acetate is an exceptionally efficient synthetic anabolic steroid. It has 15 to 50 times the androgenic and anabolic potency of testosterone. And importantly, trenbolone is the androgen that's most commonly used in these hormonal growth-promoting implants. After being implanted, trenbolone is rapidly broken down into various metabolites and excreted, and the most potent of these metabolites is 17-beta-trenbolone, which is a powerful androgenic endocrine disrupting chemical. And after excretion, 17-beta-trenbolone is often allowed to run off in the effluent of these cattle into nearby freshwater ecosystems where trenbolone has repeatedly been detected. Detected concentrations in the environment typically range from less than 1 to 20 nanograms per litre to a highest detected level of 162 nanograms per litre. And trenbolone is also relatively temporally persistent with a half-life of approximately 260 days. Now, more conventional approaches have already demonstrated the capacity for trenbolone to alter development and reproduction in a range of different non-target species. And more recent behavioural research has shown that exposure to trenbolone, even at these dilute, environmentally realistic exposure levels, can alter a range of key behaviours as well. So, for example, zebrafish exposed to trenbolone from egg until sexual maturity underwent a fully functional female-to-male sex reversal, including performing the full suite of male-typical reproductive behaviours. Exposure to trenbolone has also been shown to disrupt reproductive behaviours in female mosquitofish. And it's even been linked with altered amplexus or mate clasping behaviour in male western-clawed frogs. Despite this, we still have only a very rudimentary understanding of how trenbolone, or really pharmaceutical pollutants in general, might impact more complex, ecologically realistic behavioural processes in wildlife. And this includes, for example, possible effects of trenbolone exposure on reproductive behaviour when potential mates are encountered sequentially. And this is likely to be a far more ecologically realistic scenario in a wide array of species that live in mixed sex groups. So we wanted to investigate whether exposure to a field realistic level of trenbolone disrupts patterns, if any, of male reproductive behaviour and strategic mating effort when females are encountered sequentially. In this experiment, we used the guppy, Persilla reticulata, as our model species. Many of you will be already familiar, but the guppy is a small, live-bearing freshwater fish that's been extensively studied in behavioural and evolutionary ecology. And because of this, we have a very strong understanding of the guppy mating system and guppy mating preferences. So male guppies use two alternate reproductive tactics. They either perform courtship behaviour, where the male dances for the female to solicit copulations by showing off his coloration and ornate fins, or alternatively, males carry out what's called sneak mating behaviour for forced copulations, which essentially involves the male sneaking up from behind the female and mating with her without first performing courtship behaviour. And again, because guppies have been so extensively studied, we know that male guppies prefer to mate with larger females. And this is because female guppy fecundity, or reproductive capacity, increases with body size, meaning that there's a substantial fitness benefit to males in being choosy and preferentially mating with those larger and more fecund females. In this study, male guppies underwent a standard 21-day flow-through chemical exposure, which essentially involved males either being randomly allocated to exposed tanks that were maintained with an environmentally realistic level of trenbolone, or unexposed tanks which were identical but exposed the fish only to fresh water. The average trenbolone concentration in the exposed tanks was 2 nanograms per litre, which is actually on the low end of environmentally realistic, and we detected no trenbolone in any of the unexposed tanks. And this flow-through chemical exposure provided us with both trenbolone exposed and unexposed males, which were sequentially presented in behavioural trials with either large or small unexposed stimulus females. And you can see the different permutations of our treatments in the bottom left there. So we have unexposed males that were presented with a small and then a small unexposed stimulus female, unexposed males that were presented with a large and then a large unexposed stimulus female, unexposed males presented with a small and then a large unexposed female, and unexposed males presented with a large and then a small unexposed female. And then we had all of these permutations also where the male was exposed to trenbolone. So these behavioural trials firstly involved one male and one female being acclimated to a trial tank for 5 minutes. They were then released into the observation tank where their reproductive behaviour was video recorded for 15 minutes. The female from the first presentation was then gently netted and removed from the tank. And the second female was acclimated for 5 minutes. And this female was then released. And again, the male's reproductive behaviour towards the female was video recorded for 15 minutes. And the video footage from these trials was then analysed using an event recording software, essentially for the number of times that the male performed courtship behaviour towards the female and the number of times that the male attempted a sneak copulation with each female. So what did we find? Did males allocate their mating efforts strategically when they encountered females sequentially? And was this disrupted by trenbolone exposure? Well, first, we found no evidence that the size of a previously encountered female affected the amount of courtship or coercive sneak mating behaviour performed by males during the second presentation. So in other words, this indicates that males did not adjust their reproductive efforts strategically when they encountered different sized females sequentially. However, males from both exposure treatments, so unexposed and exposed males, conducted more frequent courting events towards larger females during both presentations. So in these graphs here, you can see presentation 1 on the left, presentation 2 on the right. In the top row of plots, you can see the number of courtship bouts performed by males towards females. In the bottom row, you can see the number of sneaking attempts performed by males towards females. And you can see that across both presentations, males performed more courtship behaviours towards larger females. And males also performed significantly more sneak mating behaviour towards large females relative to smaller females during the second presentation, although this effect wasn't seen and wasn't significant in the first presentation. But overall, these results so far suggest an absolute preference for greater female size in these guppies, which makes sense given the positive association between size and fecundity in females of this species. Importantly, however, we also saw across both presentation 1 and presentation 2 that trenbolone exposure actually caused a shift in male mating strategy towards increased coercive sneaking behaviour, while we saw no effective exposure on male courtship behaviour. And indeed, over the last few years, we've shown that this shift towards increased sneaking behaviour in male guppies exposed to trenbolone is robust and repeatable, including across a range of different environmentally realistic trenbolone exposure concentrations, in a scenario where both the male and the female were exposed to trenbolone, and even when 2 trenbolone-exposed males are allowed to compete over a female. And of course, this finding has major potential implications for both male and female fitness in exposed fish populations, including because male sneaking behaviour actually circumvents female mate choice in guppies. So why does all this really matter? Who cares about fish sex anyway? Well, there are, of course, many human-centric reasons to be concerned about contamination of the environment with pharmaceuticals, for example, the tainting of very valuable food and water resources. But the take-home message of my talk is that by altering key traits and behaviours, especially those that influence survival and reproductive success, pharmaceutical pollution can have major but often hidden impacts on ecology and evolution in wildlife populations. Now, I hope I've convinced you that pharmaceutical contamination of the environment is a serious problem, but so that this talk isn't all doom and gloom, it's important to also quickly mention that progress is being made in tackling this issue. So For example, in late 2019, an important workshop was held in Dessau, Germany, that was the first major concerted attempt to incorporate behavioral analysis into how we test for potential hazards posed by new chemicals that enter the market, including pharmaceuticals. So this was an important recent step. And even more recently, in September 2020, the European Parliament announced the first legislation that's a major strategic initiative to actually tackle pharmaceutical pollution directly. So another important recent step. And awareness is also a major hurdle when it comes to pharmaceutical pollution. So I thought I'd include a very quick plug, which is that our lab's work was recently featured in episode 2 of Connected, which is a new Netflix documentary series. So if you're interested to learn more about impacts of pharmaceutical contaminants in the environment, including on behavior, then make sure to check that out. Thank you very much for listening.
Great. Thank you for that talk. I think, Michael, you're here to take questions, right?
I am.
Okay, first of all, I would like it on the record that I care about fish sex. So
I'm glad.
Yes. Okay. So here's the first question. Do exposed males still invest as much energy into reproduction, but switch their strategy? Or do they only reduce displaying?
So the males perform an equivalent amount of courtship behavior. So the courtship behavior doesn't change. It's just that they increase their amount of sneak mating behavior. So they're performing a lot of that sneak mating behavior. And we've also, in research that hopefully will be published in the next sort of few months, found that it also affects their sperm. So it affects their sort of pre-copulatory reproduction and post-copulatory reproduction, potentially increasing sneaking and then changing their sperm quality, which sort of being played out in natural ecosystems would mean that females are being mated with potentially more by these contaminated males.
Does it make their sperm quality better or worse?
It, I guess, makes it dishonest. It changes the relationship between male quality and sperm performance.
Wow.
So it's it's complicated is the answer, but yeah, it essentially changes the relationship between their pre-copulatory traits and post-copulatory sperm performance.
Wow.
Again, making it difficult for the females.
Okay, so is there evidence for local adaptation between exposed and unexposed populations? Could exposed populations evolve to be less sensitive to trenbolone?
I wish that was the case. Unfortunately, trenbolone is an extraordinarily potent synthetic androgen. So it's used in these cows to bulk them up so that they put on mass very quickly. It's also abused by bodybuilders. So they'll use it to put on muscle mass really quickly. What that means is that even though these drugs like trenbolone are present in the environment at extremely low concentrations, even those concentrations, if they're exposed during vulnerable developmental windows, it'll actually masculinize the whole population. So you'll have 100% male populations of fish, for example, and frogs, even at very low concentrations. And so the populations don't sort of last long enough that you can see adaptation, because you'll get populations that are sex reversed. In terms of this experiment, like why look at their behavior if they're all going to be male anyway? It's because we used an extraordinarily even lower concentration. So we used on the order of about, well, on the lower end of environmentally realistic. Still, over multiple generations, you'll get a switch toward too many males, and the populations will crash, usually.
Interesting. Okay, we have a question. Do you have any ideas about the physiological mechanisms that change mating strategies? And then usually we think about higher levels of androgens like testosterone in fighter/courter rather than sneaker males.
Yeah, so we've seen all sorts of changes in fish exposed to trenbolone. So fish have androgen receptors, and trenbolone is an androgen receptor agonist. So it's a sort of a hyper. We think of these males as being hyper masculinized. They're performing more reproductive behavior. They're more aggressive. That wasn't discussed in this in this study, but we've done other studies where we see that Trenbolone exposed males will actually outcompete other males. They're more aggressive. They actually put on weight. So they do the same as the cows, the same as the bodybuilders. They put on mass relative to their length, which means that they can, in some cases, outcompete other males for females. So the whole sort of dynamics of this male-female interaction and the reproductive interactions in these populations are being thrown out of whack. Of course, this is just one instance in one species with one chemical, but what we're trying to do is use it as a representative chemical that we're saying is especially bad, like certain insecticides or certain things that you hear about all the time, to show that pharmaceuticals in the environment are a problem by using one that we know is exceptionally bad, but it hasn't been sort of played out yet. We don't fully understand what's going on in the system yet.
Okay, we have to go to the next talk, but there's a bunch more
Sure.
questions in the chat, so hopefully you can connect with those people.
I'll get to them. Thank you.
Thank you so much for talking, and definitely you can either respond to the questions in the chat or follow up by email or on the Slack channel.
Sure,
Right.
thank you.
Our next speaker is Elizabeth Tapanes, who's talking about ecological and social factors underlying pelage pigmentation in a wild Sifaka population.
Yeah, great. Can you hear me? Okay, perfect. So thanks to all of you for being here today. This is my first ASN talk, so I'm excited. And today I'm going to talk about sort of one of my data chapters for my dissertation. It's on ecological and social factors underlying hair variation in this one Sifaka population. So before I get to primate, pelage, hair, fur, I really propose that we go back, and if I get to move, and that's back to the first mammals, or the first terrestrial mammals about 210 million years ago. And it's hypothesized that these first terrestrial mammals were able to capitalize successfully on these new niches because of the evolution of the hair. This gave them protection from their environments and necessary insulation. And in the 210 million years that followed, as you probably all know, mammals have continued to diversify. And today we can see everything from these red hues to these stripes and spots to molting. And we understand that hair is really central to what it means to be a mammal. But more importantly, hair is this really cool trait to study from an evolutionary perspective. And there's many reasons for that. But one of those reasons is that hair is the product of natural and sexual selection to one extent or the other in sort of every organism that has hair. This means it's involved in physiology and signaling and everything from mate choice to camouflage. But most of what we know about the variation of this phenotype in wild natural populations comes from work in rodent populations. There are some exceptions, hares, big cats, but there's not many. And this to me is quite unfortunate because when I think of mammalian hair diversity, the clade that stands out to me the most are the primates. Primates are so incredibly varied, not just because of their colors, but because of their patterns and also because of their hair growth morphologies. As you can see, some of them even like to sport hipster mustaches. But what we know about primate hair is limited, mostly to these macro scale studies that have used images of zoo animals or museum skins. But they give us some signposts. And the first signpost is that patterns seem to be associated with measures of sociality. These are things such as group size or numbers of sympatric species in a given forest patch. And the hypothesis is that this is potentially because these patterns help individuals with identification of conspecifics or other species. The other major signpost is that melanism tends to increase in these warmer and wetter regions. And this is known as Glauger's rule. This is a very famous rule, and a lot of mammalian hair sort of follows this rule. There's a lot of debate about Glauger's rule, but a famous sort of accepted notion about it is potentially these warmer and wetter regions, they're just signaling dense forests. And these dark morphs are potentially just better camouflaged in these environments. However, within primates, Naturalists that have been going out to observe them have noted patterns that are not quite in line with these patterns in wild natural populations. And in the clade where this is most significantly noted is in the Sifaka, or within Propithecus, and now I'm really biased, but this clade is evolutionary extraordinary. They're part of a large adaptive radiation of lemurs, and they have incredible levels of both phenotypic and genetic diversity. And since about the early to mid 1900s, naturalists have been going into Madagascar and noting that there are melanic Sifaka morphs in populations of species that aren't naturally melanic. This is, for example, completely Black individuals in populations of species that are white, for example. And they noted, based on their observations alone of occurrence of these morphs, that they seem to occur in regions that are really cold. And so this goes against what we think about these warm and wet regions of Glauber's rule. And so they argue that maybe this is climatically induced selection. But unfortunately, since the mid-1900s, there's been rapid deforestation in Madagascar, and a lot of these populations where we had these Black morphs have since gone extinct. In fact, most of the species in this genus are critically endangered. However, we have one sort of remaining opportunity, and that's with the diadem Sifaka. Diadem Sifaka are still critically endangered, but they occupy this sort of large range in eastern Madagascar. And they mostly look like this individual to your right. They have these white heads, which is where they get their names from, these orange limbs, this gray back. And notably, at the southernmost part of their range, there's one really interesting population. And they kind of look like this, which is an explosion of phenotypic diversity. And so you have variations in the amounts of patterning on their faces, on their bodies, in the amount of sort of melanin on their coats or eumelanin on their coats. And we know, for example, that completely tricolored mothers can give birth to fully melanic individuals. And they live in a super interesting ecological region, and that's the Sindarivo forest. In Madagascar, Sindarivo forest is an unprotected forest, but this part of the species range is the coldest, lowest precipitation forest and highest altitude in comparison to the rest of the species range. Again, this would sort of go against Glauber's rule, if you take Glauber's rule to mean that temperature or climate tells you something about it. And throughout this region, my colleagues have been studying here for about 20 years, and they've set up 3 camps. The first, it's Matsinju. It's towards the western end. This is composed mostly of fragmented forest. Here, we also record lower rainfall, again, higher altitudes, and extremes in temperatures. There's 2 other camps that have been established. That's Ankarivori and Vatateza in the more sort of pristine forest, though there is no pristine forest in Madagascar. And here we tend to record higher rainfall, lower altitudes, and more stable temperatures. It's important to note, although we have these 3 camps in these different regions, quote unquote, as I will refer to them, the only barriers that exist here are varying levels of fragmentation. This gives us a really interesting opportunity to ask questions about hair variation at a population level scale, which has rarely been done with primates. And we're interested if these traits vary across these regions or with social factors such as group size. So as you saw, group size is known to be correlated to patterning. And so to answer this question, we really had 2 data sets. We had our main data set, which is our hair data set. This included 20 years worth of photos from about 90 individuals, half females, half males, half from each parts of the forest. And we scored their whole body coloration, pattern complexity, and contrast based on methods developed by Charlene Santana and Tim Caro. I won't get into it, but you can ask me questions at the end. And then we had a smaller data set, and this was an ecological data set. And we were really interested in if we could detect sort of significant variation across this region in climate that could provide signposts to trait variation, which we may or may not detect. And so for this, we took data from WorldClim at the population level, so from groups, and we used the P. diadema species range, which is quite large. And we ran analyses of these two data sets separately. We had principal components analysis, so 2 different PCAs, one on pigmentation and another one on degrees of patterning. And this includes countershade, which is the difference between the ventral and the dorsal surface. And we took these PC values and we plugged them into general linear mixed models with dependent variables that you can see here. And then we ran ecological niche models. This is work done mostly by my co-author, Manas Nukala. This was done with Maxent, and we assessed model performance using the area under the curve and other commonly used statistics. And what we can see right away when we look at the ecological sort of maps that are produced by Maxent, the one thing that I want you to take away from this is that, yes, there does seem to be a significant difference across these regions, and there is this west to east gradient in temperature and precipitation. This model sort of had really good support. And what you can see is that the highest 2 variables that contribute to this is precipitation of the wettest quarter and mean temperature of the coldest quarter, such that regions of the forest that are more towards the east are rainier and they're colder. It's interesting to keep these trends in mind. I'll remind you of them when we look at how color variation varies across the region. And so the coloration PCA for PC1 and PC2 accounts for about 76% of the variation here. So we're capturing a lot of variation. This is a box plot with the 3 quote unquote regions that aren't real regions on your x-axis and a significant PC value on your y-axis. And lower values are darker individuals and higher values are whiter individuals or brighter individuals. And what you can see is that the darker individuals seem to occur more towards the east or in this region of the park known as Vatateza. This region is also the coldest region, it's the densest region, and it's the rainiest region. And this was quite significant. You'll note that we still have some dark morphs that occur in the more western parts of the forest. I would like to say that it's very limited evidence, but these have been killed off by Fusa predation, and Fusa is the main predator of Sifaka besides humans. It's sort of this large mongoose-like animal. And when we look at pattern complexity, I'd like to remind you that we were interested in group size because it's known to vary with things such as patterning. And so here you can see from west to east, the 3 regions, maximum group size is on your x-axis and a PC value is on your y-axis. And here your patterning PCA between PC1 and PC2, you're accounting for about 68% of complexity or pattern complexity. Your lower values here indicate individuals that have less complex patterning, and individuals towards the top end of this are individuals that have more complex patterning, such as this arm, for example, might score a 2 or a 3, and this one might score a 1, for example. This was significant across regions and across group sizes. And this could mean that more patterning could aid conspecific communication, particularly in larger groups. And so what can we say definitively about variation across sort of the Sinjariva landscape with Sifaka? So what I can say confidently is that ecology and social factors seem to be associated with hair variation in this population. Sifakas in colder, wetter, and denser forests are darker. And this might be completely neutral and there might not be any adaptive value to this, though we're not going to focus so much on that potential story. And again, more pattern complexity in larger groups may signal that patterns are used for individual identification. And this is because a lot of individuals, not all in this population, are trichromats. And so I just want to say that we have some evidence that dark morphs have been killed off by Fusa in this population, but it's really hard to sort of say that that is because they have a greater signal detectability. These individuals also live in the smallest groups, in the smallest fragments, and it could also be that they just have nowhere to go. So there's levels of complexity here that I have a hard time teasing apart at the moment. But that said, if reasons for this variation are not neutral, I think we can consider a couple options, especially when we keep in mind Glauber's rule. And so one idea is that maybe darker forms are more camouflaged in denser forests towards the east. And this really has nothing to do with climate. For example, is it cold or is it humid? And the other idea is that potentially selection is acting on a distinct trait, which is not coloration. And so you see this with the tawny owls, where gray morphs seem to be better adapted to sort of survive these harsh winters. And you can think of it as acting potentially on cold tolerance or immunity. We know the individuals in the more eastern part of the forest have greater immunity, better immunity profiles. This could occur via a pleiotropic mechanism. For example, within the melanocortin system, you have MC1R, it's the classic coloration gene. We don't know if that's the gene that's at play here, but selection on a nearby or on a different melanocortin gene could affect coloration. And I would like to end with the fact that I essentially built an entire dissertation off of sort of naturalists' early notes. And I think that they're still important and they're still worthwhile to sort of go back to, and you can sort of build entire projects off of them. And this brings me to my next steps, and that is to consider phenotypic trends across the family. This has I've already finished this. And then we have these two other projects, the population genomics and opsin genotyping, both of FUSA and Shifox. And this has been severely paused by COVID-19. So I don't know when we're going to have these results, but they are in process. And then we want to consider other variables, such as, we have data on immunity and external parasites, and we haven't looked at this yet. And there's so many people to thank. This is sort of a project that has gone out of my hands now. But with that, I'll just say thank you, and I can take questions if you have any.
Okay, very cool. First question, great talk. In your cool result about higher complexity in larger groups, you use PC2. What was PC1? Further, I'd be super interested in hearing about how you quantified pattern complexity. We can connect about this after on Slack.
Yeah, so PC2, what contributed most to PC2 was, if I'm remembering correctly, it was body complexity as well as counter shade. So I imagine PC1 was head complexity, because those were sort of the main variables in that PCA. And yes, let's talk later about pattern complexity. I don't want to sort of deluge people in the methods.
Okay, so Elise Lauterber is bringing up the idea of inbreeding, which I was also thinking about. Can you say anything about the inbreeding status of these individuals or populations, since melanism and pseudomelanism can be related to inbreeding?
Yeah, so that's something that we are definitely considering, and it's why we're interested in doing the population genetics. What I will say is that so the only genetics data that we have is mitochondrial DNA data, and that suggests that there are the same haplotype groups, the same haplotypes in every group. So we know that gene flow is occurring throughout the forest, or we think it's occurring because of this mitochondrial DNA. We have evidence that individuals can traverse forest fragments, and the individuals that live in the fragments, they have the smallest groups, which is not what you would expect if they weren't leaving the fragments. You would expect something like the fence effect, where the group just grows larger and larger and larger. And so that's not what we see. So it is definitely a possibility. We're definitely considering it. I personally don't think that that's what's going on here. It could be a variety of other things that are also neutral, for example.
So would the darker ones then just be getting pushed to or choosing specific regions of the forest then?
Yeah, that's so the dark ones that lived in the fragments are no longer there. So they have been killed off by the FUSA. So I don't necessarily know that they are. So FUSAs kind of hunt in these sort of spurts. Like they find a shafak and they're like, there's a shafak here, we're going to kill everyone off. So it's not like they just killed the dark morph, they kill off the whole group. And so we don't really have any records of Fusa killings in denser forests. I don't know if that's because it's harder to track individuals in denser forests, or again, because there's a difference in signal detectability. But I don't think individuals are choosing sort of where to be themselves.
Okay, question. Are you sure your measure of pattern complexity is independent of your sample size, which is also your group size?
I'm not sure I understand that question.
Okay, cool. It's in the chat, so
okay. I'll follow up with that person.
I'm just reading. Okay, does group size change over time, and if so, does coloration change on that time scale as well?
Yeah, so I don't actually, after looking at it and after doing all of it, I don't actually think that group size is a great variable. So group size does change over time. In the fragmented groups, you never have a group size that is as large as in the continuous forest groups. So there are some limits to that. So in the fragmented forest, you'll have group sizes that can vary between 4 and 6, for example. And in the more continuous forest groups, your group size can vary over time between sort of 4 and 10. There's a lot of variation here. I don't think group size is a good proxy, but I do think it is signaling to us that there is something going on there, potentially with patterning or with redness, which is not something that we measured and I don't think can be measured with this data set.
Okay, we got to move on to the next talk, but thanks very much. I can see more stuff in the chat as well.
Cool, thanks.
Great. Thank you for that great talk. Our next talk, I believe, is also pre-recorded. The speaker is Kaitlyn Howell, and she'll be talking about evaluating the ecological drivers of evolved brain size differences in Trinidad killifish.
All right. Hi, my name is Kaitlyn Howell. I'm a PhD candidate at the University of Texas at Arlington. Thank you for coming to my presentation today. Today, I'll be talking about evaluating the ecological drivers of evolved brain size differences in Trinidadian killifish. Scientists have long recognized the variation in brain size across and within taxa. For example, this figure illustrates not only size differences, but structural differences in brains within the mammals alone. However, it's still unclear what drives this variation and what advantages a larger brain may confer that would be strong enough to overcome the costs of evolving a larger brain. So there's been some support for the idea that larger brains allow organisms to live longer in order to balance out delays in development or maturity. And there's also been growing support for the idea that larger brains may increase fitness in various ways. For example, one way would be by increasing cognitive processes and behavioral flexibility. But why brains may confer this advantage is still unknown. So this leads me to the central question that my research addresses. What are the ecological drivers of brain size evolution within a species? So to address my question, I work with a freshwater fish, the Trinidadian killifish, which is of course found on the island of Trinidad, which is part of a 2-island nation, Trinidad and Tobago, located just off the coast of Venezuela. So I specifically work on the northern mountain range on the south slope here, and the Arima, Guanapo, and Aripo streams primarily. So the Trinidadian killifish, Rivulus hartai, is found in various fish communities that differ in predation intensity. So upstream above barrier waterfalls, we have what we call rivulus-only sites. So in these sites, killifish are the only fish species present. They exist in high densities and thus experience intense competition. Downstream below these barrier waterfalls, we have high predation sites. where killifish co-occur with predatory fish and thus experience high mortality but decreased densities. So we know there are evolved life differences in life history traits between these sites, as well as differences in male brain size between these sites. So the males from the rivulus-only sites, the upstream sites, have evolved larger brains than the high predation males, which are from the males from the downstream sites with predators. And these differences in brain size are thought to be heritable and genetically based, given that they were maintained across multiple generations of common garden rearing. But how and why these differences have evolved is still unclear. So to answer this, I'm going to address three main questions in the rest of this presentation. So first, are there links between brain size and behavior that may be linked to fitness? Second, is there a link between brain size and fitness in the wild? And lastly, is there a link between brain size and other neurosensory components that may enhance fitness? So these questions will address potential connections between increased brain size and behavior, growth, survival, dispersal, and the neurosensory system, or parts of the neurosensory system. So first, are there links between brain size and behavior that may be linked to fitness? So to answer this, I tested the connection between jumping behavior and brain size in killifish. And I tested jumping behavior because it serves as a proxy for dispersal and foraging ability and may increase predator avoidance. So I conducted jumping trials to test if increased brain size is positively associated with increased propensity for rivulus to jump and disperse into a novel environment, and if evolved differences in brain size are associated with differences in the jumping ability of rivulus between high predation and rivulus-only sites. So to test this, I utilized wild-caught fish, both lab-acclimated and non-acclimated. So basically, lab-acclimated fish were kept in the lab for a few months before being tested, while non-acclimated fish were tested shortly after being caught. And this is actually, this was a test run while I was figuring out experimental setup, but this is essentially what this experiment looked like. So there was a small tank inside of a larger tank, and then fish were observed to see if they would successfully jump. So these figures here on the left show the log regressions between relative brain size and likelihood for fish to jump. So relative brain size is on the X, fish dispersal on the Y. In lab acclimated, so this top figure here, we saw a positive relationship between increased relative brain size and an increase in successfully jumping across all populations. In the non-acclimated fish, so this figure here, this pattern was particularly strong in the high predation fish. And this connection, the stronger connection in the high predation fish may improve their ability to avoid predators or increase the likelihood that they colonize new environments. Since we know that male fish from high predation sites have smaller brains, this connection between jumping and behavior and brain size likely does not explain the differences in brain size at the population level. So I'm also testing if there's brain size effects or if brain size affects reproductive fitness. So does a larger brain increase mating opportunities for males? To test this, I ran mate preference trials where I placed a female in the middle of a tank with one male on either end of the tank, which were housed in small clear containers with little slits in them. So females were observed for 15 minutes before males were switched sides to control for side bias in the females. And then the female was observed for 15 more minutes. And that total of 30 minutes, we calculated the time the female spent investigating each male. So after that, females were randomly paired with one of the two males for a total of 6 days, and the number of offspring they laid was counted. That was repeated with the second male. after a number of days, and again, the number of eggs excuse me, they laid was counted. So this will allow us to determine if females are choosing males with larger brains, if males with larger brains have more offspring, as well as potentially identify any other traits that females may be choosing. So this data analysis is still largely in progress, but so far preliminary results show no correlation between brain size in males and number of offspring. So brain size is connected to jumping behavior, but mating behavior is still under investigation here. And so far, what we found does not explain differences, brain size differences at the population level. So next, I examined if there's a link between brain size and fitness, specifically if increased relative brain size increases survival and growth and the likelihood for rivulets to disperse. Here, the predictions were that a larger brain would increase survival, growth, and movement. So to test this, we performed 2 mark recapture studies. The first one measured survival and growth in both sites, so both the high predation and rivulets-only sites, while the second mark recapture study focused more on dispersal and growth, so we tracked movement and growth only in rivulets-only fish. So first, the results of instantaneous growth and brain size from the 2017 study that looked at both high predation and rivulets-only sites. Here, these figures show the relationship between brain size, relative brain size, which is brain size corrected by body size on the x-axis, and then instantaneous growth rate on the y. And high predation males are here on the left with rivulets-only males on the right. So this relationship varied by population, but not by sex. And you can see a slightly positive relationship here in the rivulets-only males and a negative relation in the high predation males. So results for the females here were similar. And these patterns make sense given that increased predation is associated with decreased brain size, but all of these regressions between population and sex were non-significant except for in the RO females, which are not shown here. Growth results from the study that looked only at rivulets-only sites were similar, but the relationship between brain size and growth was stronger, although it was still not statistically significant. So we also measured survival, but found no evidence that brain size was directly correlated with survival in either the high predation population or rivulets-only population. So here are the results from measuring movement and dispersal in the second mark recapture study. So these figures show the relationship between brain size, which again is corrected by body size. So here, relative brain size has been grouped into 3 groups by based off brain size residuals. And then average net movement is on the y-axis. And basically, you can consider these categories small, medium, and large brain size. So here we have females on the left and males on the right. And we did find a strong connection between brain size and movement in these fish. So the male killifish from rivulets-only sites did tend to move more. So they actually moved about 2 times more than the females, or were 2 times more likely to move than the females. The males with larger brains were. So to summarize, we found no direct connection between brain size and survival. The only significant connection of brain size and growth was that population by brain size interaction, just meaning that growth trends varied by population. We did find that RO males with larger brains moved more from their original location. So together this suggests that brain size may help increase navigation of streams in order to increase mating and or foraging opportunities for males. So these connections between dispersal and growth may also help explain the differences in brain size that we see at the population level. So lastly, we determined if there is a link between brain size and other neurosensory components, such as eye size, that may ultimately enhance fitness. So of course, I've been talking about brain size, but we also know there are evolved differences in eye size in male killifish. So males from sites without predators, also known as these rivulus-only sites, have evolved larger eyes in addition to larger brains than the males from high predation sites, which exhibit smaller brains and smaller eyes. So here we wanted to know if specific brain components have evolved in response to differences in predation, just like overall brain size has, and if there's covariation in the evolution of brain size, brain structure, and eye size. And to address this, we utilize both wild-caught and common garden reared specimen. So here, the common garden results are shown with log transformed fish length on the x-axis and brain structure width on the y. So the open circles here and the dotted lines represent the RO data, and the solid lines and solid circles represent the high predation data. So we did find that decreased predation is associated with the evolution of larger brain structures, and this was true for all 4 of the brain structures we measured in common garden fish, so the dorsal medulla, cerebellum, objectectum, and telencephalon. So these regressions are showing the relationship between overall brain size, which is here on the left, and or brain size structure width on the right, with relative eye size on the y-axis for both. We found evidence of covariation between brain architecture and eye size, but not overall brain size and eye size. So previous work has shown that larger eyes enhance growth and survival in killifish. So this combined with our findings suggests that selection may act on specific brain structures, whether directly or indirectly, to increase visual system function, and therefore growth and survival. Also, it's important to note that we found inconsistent patterns between wild-caught and common garden fish. And this highlights that varying ecological conditions across streams may alter phenotypic expression of traits. So in summary, we found some potentially adaptive connections between dispersal, growth, and parts of the visual system and brain size in killifish. But these findings call for further research into why we see these patterns. That's all I have for today. Quickly, I'd like to thank Matthew Walsh, my PI, Shannon Baston, the rest of the Walsh Lab, the undergrads who have helped me, and my funding sources. And here you guys can find my contact information. And now I'll take any questions. Thank you.
Okay, so do we have our speaker live? Oh,
Yes.
there you go. Awesome. Okay. Given that brain size and body size are correlated, body size, which is also important for predation, dispersal, etc., could also be under selection and drive evolutionary changes in brain size. So did you look for any selection on body size?
So we have looked at like brain body allometries in terms of how, the older they get, how the brain size, how plastic it is in terms of when they're growing. So you see the most difference when they're smaller, but it does hold for almost all of the body sizes until you get to like the extreme sizes, the largest fish.
So do you think the selection on body size could be explaining some of the variation in brain size?
I guess it could be a potential factor. The body size we know varies as a life history trait as well. So, some of these fish, the ones with that live with predators, so those of course are maturing at smaller size, and then also the ones with the smaller brains. So it could be something that co-varies.
Interesting. Let's see. Is there any, so I was thinking with the jumping thing, the bigger brain ones jump more. Is there any role for biased gene flow or phenotype dependent dispersal in shaping the differences between these populations, do you think?
Can you repeat that? Sorry.
So if the ones with the bigger brains are dispersing more, could that create non-random gene flow among the populations? And if so, would that potentially explain some of the patterns that you're seeing?
Yes. So yeah, you could definitely see that, like, and this is something we've been wanting to test, you know, how if the ones with larger fish are moving more, and this was something we were going to test until COVID, so we didn't get to run that experiment. We had it set up, but never got to go back to resample for that. So yes, it could be having a factor if those fish are moving more. But we do see that, yeah, sorry.
Okay. Any hypothesis on why brain size and jumping would be related? Is jumping hard to do if you have a smaller brain?
So I think it probably, especially since we see a stronger connection in the high predation fish, I think it probably has more to do with avoiding predators. So maybe they're more behavioral, they're more flexible in their behaviors, so they're jumping more perhaps to avoid the predators, or it is, you know, jumping more to colonize and escape predators, if that makes sense.
Okay,
so that's what I'm kind of thinking.
Sorry to cut you off. Let's move on to the next talk, though, so we keep on schedule. Thanks.
Yes, thank you.
All right. Thank you so much for that great talk. Our next speaker is Abhishek Meena, who will be talking about evolution of complex reproductive investment strategies in response to aging under different intensities of sperm competition in Drosophila melanogaster.
Hello everyone, my name is Abhishek Meena. Give me 1 second, sorry. Can you hear me?
Yes,
hello?
Yes, we can hear you.
Hello everyone, my name is Abhishek Meena. Today in this, and I'm doing my PhD at University of Zurich. Today I'm presenting my master's thesis work that I did in at Indian Institute of Science Education Research at ISR Mohali in India, and in title of the evolution of complex reproductive investment strategy in response to the aging under different level of sperm competition Drosophila melanogaster. As we all know, several years ago, Charles Darwin proposed the theory of sexual selection, and he defined two forms of sexual selection: intrasexual selection and intersexual selection. And his work is mainly focused on the pre-copulatory sexual selected trait. But in the last 50 years, several studies have been shown that sexual selection also act on the post-copulatory trait. For example, sperm competition, ejaculation enrichment strategy, and also reproductive tissues. And according to the sperm competition theory, predict that the different level of sperm competition lead to the different reproductive strategy. For instance, in the male-biased condition where the high risk of sperm competition, then males try to invest more in initial mating because there is limited number of female present in their vicinity, and they have better high male-male competition in their vicinity, and they enhance their sperm competition ability, and they have better courtship behavior compared to the low level of or low risk of sperm competition. For example, in the female biased condition where low male-male competition and the relatively less low sperm competition ability, and so in this condition, when male biased condition, that females try to invest more in initial matings compared to the female biased condition, and in female biased condition, then the males know that there are enough females present in the vicinity, so they try to invest equally in each, strategically in each mating. And that's several study. I mean, previous study has been shown that support the sperm competition theory, and from one of the study from our lab, it shows that investigate the sperm competition ability in the evolving the different operational sex ratio, and they investigate the sperm offense and sperm defense, and they found that in M males, the male biased males have better higher sperm competition ability compared to the F regime males. And another study have been shown investigate the ejaculation depletion pattern in these sex biased population of Drosophila melanogaster, and they've been shown that the males from a high level of sperm competition in the MB males, they deplete faster compared to the female biased males. So in this graph, can use the laser. In this graph, on y-axis represent the progeny number, and x-axis represent the reproductive investment in each number of mating. And then they found that M males, the dotted lines is represented by the MB males, male biased males, and solid line represent the female biased. And then shows that M males deplete faster compared to the F males. And they also shows that it also reflects in the reproductive tissues for accessory gland and the testis size. And so so what we know from these data from previous studies, that M males have increased their, in young males, M males have the higher sperm competition ability compared to F males, and M males invest relatively higher in initial mating compared to the F males. And it suggests that the selection regime play important role in the post-copulatory sexual post-copulatory traits. However, it is still less clear whether the selection can enhance the post-reproductive traits in the later age. So we propose a hypothesis to investigate the differential level of sperm competition is correlated to the reproductive performance in the response of aging, and we ask following questions: does the male aging age and the selection regime affect the ejaculation investment, and does it also reflect in the tissue? So to answer this question, we use laboratory adapted sex biased population of Drosophila melanogaster, and we have these population are derived from the one common LHST ancestor LHST population, and we drive in 3 different selection regimes. In male biased condition, where 3 males and in ratio of 3 males and 1 female condition, and in female biased condition, where 3 females and 1 male condition in ratio of 1 male condition, and control is just a control equal number of male and female individual. And these population are maintained around in our lab around more than 250 generation of the selection pressure. So we got to answer this question, I collected egg from M regime and F regime males, and we collected 9th on 10th day we collected virgins, and then we randomly assigned 3 category, 3 age classes. Young age is 12, basically 2-3-day-old males, and middle age is around 14-15-day-old males, and old age male is 24 and 25-day-old males. And these age class are defined based on the young age. We applied a selection pressure on young age, and old age is basically when the reproductive fitness is declined and the age of that after 25 days of old flies, and middle is the intermediate between these stages. And the females are collected from the common ancestor population, that is LHST population, and we are generating the similar way as we generated the males, but we always use young female for all these different age assays. So we collected virgin males and females, and then on the day of experiment, we combined 1 male, 1 female, and in a vial, and in a extended vial, and then after each mating, with males transfer into after mating, females transfer into the oviposition vial for the egg-laying window, and then next new virgin female is introduced to the focal male to the mated, and then we perform again second mating, and so on. We perform 4 consecutive mating in these males at 3 different age, and we also dissected some reproductive tissues as a proxy of investment. So we use thorax size as a normalizing body size, and we took the area of gland area, testis area, and the testis length with 3 age groups and with respect to the mating. And what we found that this graph is represent x-axis is represent the mating number, and on y-axis is represent the total number of progeny that laid by in initial 24 hours of egg-laying window, and the the red line is denote for the young age, green line is denote for the middle age, and the blue line is represent for the old age male. And what interesting, we found that young male show clear depletion pattern with with respect to the number of mating, but middle and old age males produce equal number of progeny in each number of mating, and it might possible because the sexual selection pressure applied only on young age males, only at young age stages, and we found that there is a significant effect of the mating number and age, and the interaction of mating number and age, and we found that M males are produce more offspring compared to the F males, but there is no we don't find any clear reproductive strategy in M and F males. And similar trend we also found in the after 24 hours of egg laying window, the next 48 hours. So and we and in this graph we also we investigate the SSC gland area. On y-axis in the SSC area, and on x-axis represents the mating number. 0 is a virgin male as a control, and 1 first mating and 4th mating. And what we found that SSC gland area is increased with aging, and it declined with the number of mating. And mating number and age we found a significant effect of mating number and age on them, and there is an interaction between mating number and age. And we also investigate the testis area, and testis area is we found a similar trend in this with respect to the progeny data, and we investigated the testis length, and we found that the testis length is shorter in the young age, but it increased with age, and it declined with the number of mating. So what we conclude from our talk is we did not find any reproductive strategy in these sexual selected regimes, even after 250 generation of maintaining in the lab. And the population had different the interactive effect of the aging and mating number on the reproductive investment pattern, which is unique in the trade considering. And among the pattern, we found the main effect is mating number and age. And their interaction is very consistent throughout the study. And we from this study, we suggest that the reproductive performance is not similar throughout their lifespan. So it affects with the age and mating number. And I would like to acknowledge my supervisor, Dr. Anji Prasad, and and Dr. Manjri Jain for their providing a microscope facility, and my all evolutionary lab members. And thank you so much. And if you have any question, yeah, we will last.
Okay, great. We have lots of time for questions. I think people are still typing. So I'll ramble a little bit while people are still typing. So I don't know if you were here for the very first talk of this session from Vince Formica, but also found patterns where essentially males get better as they get older in another insect. Males and females get better at reproducing successfully. And Vince, please feel free to jump in if I'm misconstruing or misremembering your results. So I'm so fascinated by this contrast because flies are not considered a long-lived insect probably in the wild, right? They're probably not selected to be awesome at mating when they're 24 days post-eclosion. So I don't really have a question. I was just interested in starting a discussion and maybe if Vince is still here, he can chime in as well about these changes over age and what they mean and how they might evolve.
Sorry.
Just any thoughts about why the males might be improving their reproductive performance with age, especially since they might not be living that long in the wild?
Yeah, in the wild, they are not living that long and not that selection pressure, but the selection pressure is applied on these conditions. These are the laboratory adapted population that manipulate in the lab condition, and but that selection pressure only applied in the early age, and it also influences when the selection is going on with the aging. It also influences that the males that evolve in the male biased condition, they have better sperm competition and their fighting ability. So I thought that is also reflect in the older age. So they have this also perform better in the older age. But we haven't find any differences in the old age, because only we in these population we just maintain on the young age, and then we just discard the flies and start the next generation. But in wild condition is very unlikely. I agree with you, but very unlikely to find any old flies in respect of selection pressure. Yeah.
So Becky Fuller's bringing up that males may increase sperm production as they mate more. So mating itself might increase sperm production. Does this happen in fruit flies? And could this possibly account for the effects in the young males or the difference over age?
Right. Yeah, that mating is increased with the sperm production, but we first thing we just perform we just as a proxy of sperm number, we use a progeny data. So just a progeny, because we don't have GFP and RFP flies, so we don't know whether the sperm they invest more sperm, transfer more sperm in or less sperm in each mating. We don't know whether thing, and we just as a proxy, just initial proxy as a competitor of the sperm number, and what else? And yeah, but mating, but with increasing mating number, the sperm production is also increased. But that when tested, when males get recover after each day, so we perform mating each separate day. So this experiment perform consecutively mating, so it's not increase. So they decline their testes. That suppose they have 50 sperm, and then each mating the sperm number is decline basically. But the people have been shown that mating number increase their sperm production. That is only happen when we perform single mating in each day, and then next day again male perform better reproductive fitness, and each day they have higher fitness, but not the one day. But we perform on the experiment on the single day with 4 consecutive matings. So
time dynamics. So if you had males that you just aged to be 24 days old and never tested them until that day, do you think you might get different results?
Right, because it might possible, because the sperm become older and older if they're not mated any male, and they decline their fitness. And but if mating is also if they're continuously mating, then the sperm they produce new sperm each day, and then they're better in quality and produce better offsprings. But we use virgin males as a gesture, but that also decline the sperm quality of the sperm with the age.
What do you think is the role of seminal fluid proteins? This is from Giuliano Morimoto.
Seminal protein, the role of seminal protein, they basically like suppress that not the another male is not going to mate, so they defends the female to remate basically. So it's not allowed to remate. The males, those have better sperm, they transfer more sperm, seminal protein fluid, then they have more remating latency basically. So they are not mating anymore. The males have defense ability. So the sperm seminal protein avoid the remating basically. So they enhance the reproductive.
So does that mean, do you think that those seminal proteins did not play a role in your study then, since?
Yeah, now in this study is not a play role, but in general they also transfer during the mating. They transfer sperm as well as the seminal protein, but the seminal protein play an important role when females have get another mating opportunity. Then the previous male have better seminal, like transfer more seminal fluid. Then the female have declined the remating basically. Yeah.
I mean, I think some of the seminal proteins can also affect things like female ovulation and lots of other female behavioral.
And there is other role also. Yeah.
So so it's surprising that the sex ratio manipulation didn't seem effects, which would be cool. Do you have any plans to test these males with the females that co-evolved with them in the sex ratio manipulation populations?
I not any plan yet, but maybe some other else. Like I finished my master's and now I'm starting my PhD in different lab. So
congratulations.
yeah, they have only that lab have these sexual selection lines. So they maintain around 250 generation in the laboratory condition. And here we have different flies. So it's quite hard. Yeah.
He's saying come to Aberdeen. Maybe after COVID. Okay, great. Thanks.
All right, thank you so much for your talk. We're moving on to the last talk of the session before the break, before we have a 20-minute break where I presume there's going to be lots of coffee and cookies, depending on where you are. Our last speaker before break is Renato Nali, who will be speaking about male aggression and its evolutionary associations with territoriality and site fidelity in frogs.
Hi everyone, thanks for coming to my talk. My name is Renato and today I'm going to be presenting some results with my colleague Angelette Valencia Aguilar on male aggression and evolutionary associations with territoriality and site fidelity in frogs. Aggressive behavior is actually a form of communication, so it can be used across animals with different functions. For instance, to end disputes over resources, territory or food, or even mates. In this case, it's used to increase reproductive potential, so mates are actually resources. And it's also used to escape threatening situations, for instance, when fighting against a predator. In frogs, aggressive behavior is very much related with breeding patterns and breeding behaviors, and it can be escalated, and basically of two types. So it can be a non-physical aggressive behavior, which is visual and acoustic signals, for instance, body postures and limb movements, like here, and acoustics such as different types of calls, like aggressive calls. And it can be also physical. For instance, it's in male-male wrestling, which could be pushes, bites, bumps, and any type of physical interactions among males, as you can see here in these two photographs. Aggressive behavior in frogs is related with the defense of specific resources within an area, and it is related normally with females or the acquisition of oviposition sites, and also for male interspacing in a reproductive coral. So aggressive behavior is also used by males to maintain spacing among males in the reproductive corals. One interesting thing is that in frogs, aggressive behavior can be elicited when another male invades the resident space. So if you think about space, there's two concepts that are important here. It can be related with aggressive behavior. For instance, territoriality and site fidelity. So territoriality is defined as an individual holds an area employing stereotyped signals or agonistic encounters to defend it. So in territoriality, the individual actually defends this area. On the other hand, site fidelity, an individual remains in a particular area for a period of time, can be days or weeks. For instance, a male calls on the same leaf night after night. So it is site fidelity but not really territoriality because the male does not defend the area. So that's site fidelity. And considering that frogs show elaborate and conspicuous social behaviors, including different types of male aggression, here we aim to answer two different questions. So, how did physical and non-physical aggressive behaviors evolve in this group? So, what were the evolutionary transitions? And also, are those traits correlated with the presence of territoriality and site fidelity in frogs? So the fact that they are aggressive or not, is it correlated with territoriality or site fidelity in frogs? So we collected data from the literature and also personal observations of frog species regarding the type of aggression. So here you can see a cool color green and then warmer colors yellow, orange and red, so that means an increase of aggression. So we had four types of aggression in frogs. First, non-aggressive, which means for this species there's no observations in long-term studies or many studies for a single species about aggression. So we consider that this species was not aggressive. There's non-physical aggression only when species displayed acoustic or visual cues, for example, aggressive calls, changes in call parameters that the authors clearly state that they were related with aggression, and also body postures, limb movements, chasing, as you can see here on the right in this video, a male calling and also doing limb movements. Also, we had physical aggression, which we also call combat, which would be pushes, kicks, bumps, bites, male dislodgement, any type of physical interaction between two males, as you can see here on the right with a video of a fight, a male-male fight. And if a species had both types of aggression, non-physical and physical, we characterized as such, so both types of aggression as here. We also search for territoriality and site fidelity in different species, so we characterize these behaviors as presence or absence. For site fidelity, we used mark recapture studies or robust natural history observations of remaining in the same area without any types of defense, so that would be site fidelity. And territoriality also as presence or absence, as stated by the author. So if the author stated that the species is territorial and why, we consider that, and if the author didn't mention anything specifically about territoriality, we also consider territorial if the species presented site fidelity and also any type of aggressive defense to that area. So you're probably thinking that absence is not really robust because it can mean that this behavior was not observed yet. We are aware of that, but we had a smaller data set with species that had actually natural history studies from this species many times, many studies for the same species, so we are a little bit more confident about the absence. So in those studies, nobody had observed any of that, so we think that it is more reliable. For our analysis, first we did evolutionary transitions of aggression, so we reconstructed these 4 states of aggression, so no aggression, non-physical aggression only, physical aggression only, and both types of aggression. And we tested 3 models of evolutionary transitions, the equal race, the all race different, and the symmetrical model. We chose the best model and also plotted these results to see the transitions and what they were telling us. For the analysis of physical and non-physical aggression against territoriality and site fidelity, we ran correlation of discrete characters analysis, and so we had 4 analyses for this, and for each of them 4 possible models. So the independent evolution, which is no correlation among the traits, and the first variable is dependent on the second is dependent on the first, or they are both interdependent. So in each case, the only model that is not, that shows no correlation is the independent model, and the other 3 show some type of correlation. So we chose the best one. And we chose the best significant model with lower AICC. And for all this analysis, we used the most complete and updated amphibian phylogeny, which is Yates and Pyron 2018, and we pruned the 3 accordingly. So we gathered a data set. We started with 2 previous data sets from 2 of our studies, these 2 studies here, which helped us find other articles for the specific characters that we wanted. So we looked about 300 articles or academic works like theses and dissertations, and we ended up with a set of 178 species from 33 families with most of the information that we needed. So our analysis ended up having a range from 160 to 178 depending on the analysis and the information that we needed for each analysis. And here I'm going to show some maps of the variables just for a first look. Here is the 4 states of aggression with the same 4 colors that I showed you before. And we can see here in this mapping is that both types of aggression is the most common form of aggression in frogs, and that they are distributed across the tree. And also the other 3 types, which is non-physical only and physical only, they occur across the tree. So it's not really a pattern of more basal and more derived states, rather it's a pattern of distribution across the tree. Here you can see site fidelity, so presence is blue and absence is red. You can see here that it's also distributed across the tree. There's not a clear pattern of more basal or more derived states. whether it's distributed across the tree, which also happens with territoriality. You can see here that both states are distributed across the tree. We're going to analyze that later. So here's the analysis of evolutionary transitions of male aggression. You see the 4 states and the colors they represent. And the best model for that was the all rates different, which means that each arrow represents different evolutionary transition rates. And actually the thickness of each arrow is proportional to the transition rates. And first we see that both types of aggression is the most common type of aggression in frogs, but here it's a fairly high evolutionary rate in which absence of aggression evolved directly both types of aggression. So there's not really a path in which absence of aggression would evolve non-physical aggression, then physical aggression, or both. So this is non-existent here. So it's directly from absence of aggression to both types of aggression. Another interesting result here is that from species that have non-physical aggression, they are much more likely to lose this type of aggression than to gain physical aggression and have both types of aggression. So this all shows that aggression can be is not stable. It's not a stable behavior across the evolution of frogs, and they can be advantageous or not, so they can be lost or not, and that may as well be related with site fidelity and territoriality, and that's why we ran this analysis as well. So now I'm going to show our 4 results on the correlation on combat or non-physical aggression versus territoriality and site fidelity. So these graphs here represent pairs of traits. Whichever is blue means presence, and whichever is red means absence. And here you can see that there's different arrows which represent different evolutionary rates. Okay, so the first result that we had was that combat depends on territoriality. This means that if a species is territorial or not, that would represent different evolutionary rates. And of course we have many results here, but I want to draw attention to this one. So if a species is territorial here, they gain combat multiple times faster than they lose it. Okay, so if a species is territorial, combat is an important trait, which shows an advantage in maintaining those territories. About non-physical aggression, they are and territoriality, they are interdependent. So we also see that depending on the territoriality and physical aggression, the evolutionary rates differ. Okay, and here I want to point to this result here. So if a species is non-territorial, what happens is that they lost non-physical aggression 3 times faster than the opposite. So for a species that is non-territorial, it's not really an advantage to keep having non-physical aggression, so they tend to lose it. So non-visual aggression, non-physical aggression like calling and visual behaviors may also be costly. For calls, we know that they are very costly and might as well be the case for visual aggression. So this shows that aggression is not advantageous every time. Regarding site fidelity, we found similar results, but here we see that combat depends on site fidelity, but as you can see here in the arrows that evolutionary rates are lower than what we found for territoriality. But in any case, the tendency was the same. Here, for species that have site fidelity, they tend to gain combat multiple times faster than lose it, showing that combat is important also for species with site fidelity. We also found that non-physical aggression and site fidelity are interdependent here. And I want to draw attention here to this result, which is for a species that shows non-physical aggression, what happens is that it presents site fidelity much more than the opposite. So here it shows a clear advantage of non-physical aggression also in maintaining sites. So for site fidelity, non-physical aggression is also important. So when frogs fight, they actually sustain different types of injuries, like here with the scars, as you can see here, or males may even fight to their death. So it is hypothesized that non-physical aggression would be a more derived strategy because it would allow males to keep their sites, their territories, without sustaining the injuries that come from physical combat. But here we found no evidence for that, so we didn't find that non-physical aggression is a more derived strategy to avoid injuries, because most species across the tree displayed both types of aggression. However, here we're thinking about presence and absence of, because that's the type of data that we have available for dozens of species in the literature, but the frequency of each behavior would be more accurate than presence and absence. So perhaps the more derived strategy would be less combat and more non-physical aggression. So combat would be a last resource only, and it wouldn't be about if a species has the ability of doing this or that, but how much they invest on each type of behavior, so that remains to be tested. I also wanted to bring this to discussion because I told you that aggression in frogs is very related with breeding. And in frogs we have basically two breeding patterns, which is the prolonged breeders and explosive breeders. So in prolonged breeders, aggression is costly because they have to reproduce over several weeks or months. But it becomes advantageous when maintaining important sites, for instance oviposition sites or access to females. So we do observe that in prolonged breeders, aggression is also advantageous. In explosive breeders, aggression is still costly, of course, but they do have fewer opportunities to obtain females. So in this case, they do not have territories, but rather they search actively for females and also amplexuses, in which they dislodge males from amplexus. As you can see here, a female, a male, and another male try to dislodge. So this is a type of aggression as well, but it is important because the aggression occurs differently in these two breeding patterns. And also explosive breeders are much less studied here because it's difficult to find breeding events in the field compared to prolonged breeders. In our data set, for instance, we have 20% of our sample of explosive breeders. So we are collecting more data and trying to solve this, but it's interesting to see that the pattern may be different if we consider prolonged and explosive breeders. So in conclusion, both physical and non-physical aggression are correlated with the ability of male frogs in maintaining important sites, but aggression likely comes with high costs and can be either lost or gained throughout the evolution, depending on the advantage conferred by it. So we have here, this work shows clearly these two sides of aggression. It can be advantageous or not. Well, thanks for watching. I'm sorry I can't be present live here, but if you have any questions or suggestions, because it is an ongoing project, you can email me and I'll be happy to answer your questions. Thank you.
All right.
So I think the speaker indicated that they're not present here on the call. Is that, speaker, if you're here, can you? Okay, pretty sure they're not here. So great. So we can go to break a little bit early, which we probably all need.
Awesome. All right, and no co-authors either, right? Okay. So it's time for break. We will resume in about 20 minutes. What is here on the West Coast is 2:40, and you can adjust accordingly. I thought if people are interested, if we were in a Silmar, we would be probably going to and looking at the ocean. I have some photos of elephant seals from Año Nuevo, which is right down the road from a Silmar. So I can put that on my screen and share that while people are on break, just for fun and for just background. Again, go get up, stretch, get coffee, cookies, rest your eyes, and when you come back, there will be some elephant seals on the screen for you to enjoy.
Great, and thanks to all the speakers in this session. It was great.
Right. Thanks, Julia. Thanks for everyone. See you in a few. Wooder, is it okay if I share the screen?
Yeah, go ahead.
Okay, welcome back everyone to the social behavior session of the meeting. This is the second half of our session, and we're starting off with Mitch Daniel, who'll be telling us about experimental evolution reveals extreme negative frequency dependent sexual selection on male guppy color pattern.
Hello, and thank you all for tuning into my virtual talk. I'm currently a postdoc at Florida State working with Kim Hughes, and I'm going to be sharing some results from an experiment in progress in which I'm using experimental evolution to ask why the color patterns of male Trinidadian guppies have incredibly high levels of polymorphism. The paradox of variation is a classic problem in evolutionary biology. Populations often exhibit much higher levels of genetic variation than can be explained by standard models of mutation, drift, and directional selection. And this genetic variation, of course, has fundamental implications for many evolutionary and ecological processes, including the evolution of disease resistance, responses to environmental change, and population persistence. Consequently, understanding how genetic variation is maintained remains a central question. One particularly powerful process for maintaining genetic diversity is negative frequency dependent selection, which I'll be referring to as NFDS. This is when the fitness of a phenotype is inversely related to its frequency in the population. Because rare alleles are favored, they're unlikely to be lost from the population, thereby preserving genetic variation over evolutionary timescales. Some famous polymorphisms maintained by NFDS include color variation in the elder-flowered orchid, the 3 color morphs of side-blotched lizards, and the shell polymorphism of grove snails. But in the last few decades, examples of negative frequency-dependent selection have become relatively common and now span a fairly diverse array of taxa, including the examples that I'm showing here. This literature suggests that NFDS may be of broad importance for the maintenance of genetic variation in ecologically relevant traits. However, there's still much that we don't know about the causes and consequences of NFDS. In particular, relatively few studies have identified the ecological processes that drive NFDS, even for taxa in which NFDS has been reported. And this, of course, makes it difficult to predict how general NFDS is likely to be across different species and different kinds of traits. And in addition, most examples of NFDS come from studies that have only looked at selection acting on a single generation. So the consistency of NFDS and its capacity to maintain genetic variation over multiple generations is something that has received much theoretical consideration, but has rarely been empirically characterized. And my project is an attempt to help address these 2 gaps in our understanding of NFDS. To do this, I am using the Trinidadian guppy, which is an excellent study system for looking at negative frequency-dependent selection for a couple of different reasons. So first of all, the color patterns of male guppies are one of the most extreme examples of morphological polymorphism. So these 6 males were all sampled from the same tributary and reared in the same environment. And as you can see, they exhibit this high level of heritable variation in color pattern. The guppy is also a great study system because previous work has identified 2 ecological mechanisms that may contribute to the maintenance of this genetic variation, and both of these mechanisms have been observed in natural populations. So males with rare color patterns have a survival advantage, and this is probably the result of predators being more efficient at preying on common morphs. And males with rare color patterns also have a mating advantage, though it's not entirely clear what's causing it. And my project is focused on understanding this latter mechanism, the rare male mating advantage. And I'm doing this by asking 2 related questions. So first, is sexual selection alone capable of maintaining color polymorphism over evolutionary timescales? And second, is the rare male mating advantage underpinned by female mate choice and/or male-male competition? Answering these questions will help to advance our knowledge of NFDS in guppies and to address those broader gaps in our understanding of the causes and consequences of NFDS. And I'm asking these questions using experimental evolution. So I've established populations of guppies in the lab that varied in the initial frequency of 2 different color patterns. Each pattern was initially rare in half of the populations and common in the other half. And this allows me to disentangle frequency-dependent selection from any directional selection that might also be acting on these traits. If there is NFDS on color pattern, then I expect the initially rare color pattern to increase in frequency. And if given enough time, it should approach some equilibrium frequency at which the two phenotypes have equal fitness. To test this prediction, we need to measure the frequency of male color patterns. But these color patterns are complex and have many axes of variation, so we need to simplify things in some way so that we can measure frequency. And I'm using isogenic lines to do this. So these lines have different Y chromosome haplotypes with a homogeneous genetic background. Some parts of color pattern, particularly orange and black spots on the body, are strongly Y-linked. So these lines give us distinct color pattern features that we can identify to track their frequency. Now, these lines were founded by sampling males with these Y-linked patterns from a natural population, and there is a lot of within-line variation in other aspects of color pattern, particularly on the caudal fin. And what this means is that the color patterns we are using and the variation in these patterns should be not too different from what we see in natural populations. To further mimic natural conditions, each population consists of 100 adult males and 150 adult females housed together in a 300-gallon mesocosm. And these tanks are lit by full spectrum lights that simulate sunlight, and they're lined with gravel to imitate a natural visual backdrop. And I'm regularly culling individuals to create discrete generations and to maintain density and sex ratio throughout the experiment. In addition to varying the initial frequencies of our two isogenic lines, I'm also manipulating the opportunity for sexual selection to operate. So in 8 of the populations, the fish are able to interact and mate freely with one another. And in 2 of the populations, I'm minimizing sexual selection. So every generation, I assign matings by housing each male with 1 or 2 females. Guppies have internal fertilization, so after allowing time for matings, I can then transfer the fish to the mesocosm with males on one side of the divider and the females and eventually their offspring on the other. This procedure prevents female mate choice and male-male competition, meaning that these divided populations provide a control for the effects of sexual selection. So if sexual selection alone can promote genetic variation in color pattern, then we should see an increase in genetic variation, meaning that the phenotypes evolve towards more balanced frequencies in the free interaction populations, but not in the divided populations. All right, so here I'm showing how the frequency of the initially rare allele on the y-axis has changed across generations, and I've found patterns consistent with our predictions. So in the populations with free interaction, the initially rare phenotype has rapidly and consistently increased in frequency across generations. And it looks like the frequencies may be starting to level out as they approach 0.5, though we don't currently have much data for that part of the range. And this trend appears to be driven predominantly by sexual selection, because in the sex-divided populations in blue, the phenotypes have remained much closer to their initial frequencies. And indeed, I found a significant interaction between generation and treatment, indicating the presence of negative frequency-dependent sexual selection, or NFDSS. These patterns have emerged over about 6 generations, showing that sexual selection is capable of rapidly increasing genetic variation in male color pattern. All right, so having observed this rapid response to NFDS in the populations with free interaction, I next wanted to estimate the strength of NFDS on color pattern. For any given phenotype frequency, NFDS will resemble directional selection. And that means that we can use a model of directional selection to estimate how selection is acting on each generation in each population. And then by examining the relationship between the strength of selection and phenotype frequency, we can infer something about frequency dependence. And since we're looking at features of color pattern that are inherited in a Y-linked manner, we can use a simple haploid model to estimate the strength of selection, S, from phenotype frequency changes. All right, so here I'm showing the relationship between generation on the x-axis and the strength of selection favoring the initially rare allele. And each dot is the strength of selection for one generation in one population. All right, and I'm looking first at the populations with free interaction. So in the initial generation, we can see that the average strength of selection is about 2, which means that males with the rare color pattern are siring about 200% more offspring on average compared to males with the common color pattern. So we see very strong selection favoring the rare allele or the rare haplotype in early generations, and then selection approaches 0 in later generations. And in the control populations, in contrast, selection hovers close to 0 throughout. Now, these trends are not statistically significant, probably in large part because the estimates of selection are quite noisy compared to the frequency data. However, the trends are consistent with negative frequency-dependent sexual selection, and it's possible that a clearer signal may emerge as we collect a more robust sample size. All right, so next, next I'm showing the mean strength of selection favoring the rare allele or the rare pattern in the free interaction populations. So basically I've taken the average of all those red dots. And as you can see, mean selection is 0.75 and is significantly greater than 0, suggesting very strong NFDS. And this measure is probably quite conservative because it includes generations in which the initially rare allele had reached a frequency close to 0.5. For the sex-divided populations, the average strength of selection was much closer to and was not significantly different from 0. So this pattern is again consistent with very strong negative frequency-dependent sexual selection on male color pattern. Collectively, these preliminary data suggest that sexual selection on male color pattern is a potent evolutionary force that's capable of rapidly promoting genetic variation in color pattern. So next, I wanted to determine the ecological processes underpinning this negative frequency-dependent sexual selection, specifically whether it's driven by female mate choice and/or male-male competition. This is a question that's been partially investigated by past work. So several previous studies have shown that female guppies have a mating preference for males with rare or novel-looking color patterns. This preference is known to have consequences for male reproductive success, and so it could be responsible for NFDSS. Past work has also tested whether male guppies direct higher levels of competition towards males with patterns similar to their own. This competitive strategy could generate NFDS, though in a previous experiment I found no evidence of it. However, one limitation of this past work on both female mate choice and male-male competition is that it examined the behavior of individuals that developed within relatively small tanks and social groups. So I wanted to ask whether we see similar results when individuals develop in the more realistic environment provided by the mesocosms in this experiment. So every generation, I'm taking 10 females from each of the populations with free interaction, and I'm measuring their mating interest towards one male from each of the two lines. As a reminder, in the first 3 generations, the initially rare color pattern was still quite rare. And in these generations, females displayed a significant preference for the initially rare pattern, which is the one shown in green here. And in generations 3 through 5, when the initially rare pattern was closer to a balanced frequency, this preference was no longer significant. So this change in preference is consistent with female mate choice being the mechanism underpinning NFDS. And these results also indicate that female mate choice does respond to the frequency of color patterns in an ecologically relevant physical and social environment. The experiment testing whether male-male competition can maintain color polymorphism is being conducted by Alexa Guerrera, a graduate student in the lab. So she has tested the competitive behavior of males from the first generation of these mesocosms. And Alexa's preliminary data suggests that males direct more competitive behaviors towards rivals with a different color pattern from their own, though this trend was only significant for one of the two lines. This pattern runs counter to our predictions and should erode genetic variation in color pattern rather than maintain it. So together, the data from these behavioral experiments suggests that negative frequency-dependent sexual selection is primarily the result of a female mating preference for rare color patterns, and that female preference maintains polymorphism by overpowering an erosive process of male-male competition. Okay. So collectively, the data that I've presented today provide preliminary evidence that female mate choice exerts very strong NFDS on male color patterns, and that this explains the rare male mating advantage and extreme color polymorphism that's been observed in guppies. These results also have implications for our broader understanding of the causes and consequences of NFDS. A mating preference for rare or novel phenotypes has been observed in a small but diverse set of organisms. Some of these organisms exhibit conspicuous polymorphisms, and others may exhibit polymorphisms that have so far eluded human observation. Published tests of mating preference for novelty remain relatively rare, so it's possible that this kind of a preference is important for maintaining genetic diversity in many other taxa and traits. So with that, I'm grateful to the members of the Hughes Lab for their feedback on this project. Many graduate and undergraduate researchers also helped with data collection and maintaining these fish populations. Thank you to our funding sources and to you for your attention, and I will be happy to take any questions.
All right. Thank you so much for that lovely talk. We have our first question here. Male color patterns are so complex. How do real guppies in nature make choices about which rare aspect of color pattern is important to them? Depending on the trait, most males can have some rare aspect of color pattern.
Yeah, I think that's a great question. And definitely anecdotally, in a lot of natural populations, almost every male has a color pattern that's distinct from all the rest in some ways. And we're not entirely clear what criteria females are using to, assess how rare or how distinct a color pattern is. So one of the things that we've done in collaboration with Jen Valvo, who's a former PhD student in the Hughes Lab, we've developed a pipeline for taking basically these highly multivariate measures of male color pattern. And then we can use sort of multivariate statistical techniques to try and calculate like dissimilarity indices among males. And that may be one way to sort of get a sense of, at least sort of in terms of objective color pattern, how different are males from one another. And then hopefully from there, we can try to relate that to female preference in some way to sort of get a window into what's going on in the brains of these females.
Great. Another question. Do you think the shift in female preference over the generations were due to evolution and/or plasticity?
Yeah, that's another great question. It's not really clear. So one of the things I want to do with so we're planning to run this experiment for a total of 10 generations. And one of the things I'd love to do at the end is some type of common garden experiment to have a look at whether or not female preference has evolved over the course of this experiment. Because there is some theory suggesting that you can get the coevolution of this female preference for rarity and the frequency of these male color patterns. So there's a reason to think that that might be going on. So I'd definitely like to look into that further.
Wonderful. I'll ask a quick question. Oh, sorry, no, someone already typed in. Do you think the sperm storage capacity in dead males have higher reproductive success is interacting with your results?
Right. I don't think it could be having too much of an effect on the results in this experiment because we were enforcing discrete generations. So I was periodically culling all of the adults from the population. And so females would have only been able to go through, I think, two to three reproductive bouts, depending on how quickly they started reproducing. So I don't think, and mortality rates were quite low in our mesocosms. So I think the posthumous male reproductive success is probably not happening very much.
Right. We have time for maybe another one or two questions. Let's see if something pops up. Well, while we wait for people to type, I'll ask you quickly. So I think a little bit related to the first question you got is how would you, is there any difference in the predation risk of the two phenotypes, and how would you add, if you add other factors other than mate choice, how would that influence your results?
Yeah, so I don't know if anybody's quantified predation risk working with these particular isogenic lines. So I don't know. There's some evidence showing that predators have a preference for going after males with color patterns that are similar to the ones they've seen before. But I think that's all we really know. I don't think we know much about quantitatively how rarity relates to predation risk, if that gets you your question.
All right. I think we have time to transition to the next talk. Thank you so much for the lovely talk and for answering all the questions.
Thanks.
Okay, our next talk will be from Liz Lange, and she'll be telling us about female-female social bonds in baboons.
Just trying to find the unmute button. Okay, can everyone see my screen? I notice everyone's not looking at me, but I'm gonna just assume you can. Yep. Thanks, Noah. Okay, great. So I'm going to tell you a bit about my postdoc research, which focuses on the relationships between early life adversity, adult phenotypes, and survival in baboons. So understanding the factors important to differences in fitness is a key question in biology. So for example, we know that developmental and environmental processes during early life can affect adult phenotypes, and that adult phenotypes can affect fitness, which is often measured by reproductive success and or lifespan. And of course, differences in the environment, whether that be abiotic, biotic, or social, can affect both early life and adult phenotypes and ultimately drive fitness. Often studies focus on one part of this causal chain, for example, determining the relationships between early life and adult traits, or determining how early life affects fitness. But it's unclear how these processes are linked in many systems. For example, from many studies in humans, we know that early life experiences are important. Adverse childhood experiences like abuse, neglect, and household dysfunction can negatively affect developmental processes, and ultimately this has been shown to lead to negative behaviors and reduced lifespan in humans. And of course, this is not just a human problem. There are many different animal systems who we may appreciate or like to think about more than humans, and early life experiences have been shown to have important and lasting effects in a variety of animal taxa. However, it's unclear how experiences during early life and adulthood translate into differences in lifespan in many species. One hypothesis to explain how early life adversities lead to decreased lifespan is the biological embedding hypothesis. And this hypothesis predicts that adversities during the early life create a pro-inflammatory phenotype, which promotes mistrust in others and ultimately makes individuals have less social connections to others during adulthood. In addition, this pro-inflammatory phenotype exaggerates the stress response and leads to increased glucocorticoid levels. These adult phenotypes are then hypothesized to lead to negative health effects that ultimately decrease lifespan. And while we hypothesize that adult phenotypes drive the relationship between early life adversities and lifespan, this is yet to be explicitly tested. For example, we don't know if early life adversities affect fitness directly, as shown by this black arrow, or if there's a mediation effect where early life adversities affect adult traits, which in turn affects survival, as predicted by the biological embedding hypothesis. And of course, all of these effects can be modulated by the environment. And for now, I'm not going to talk too much about that, but I'm happy to talk later. For now, I'm just going to take the environment into account with covariates. In the Amboseli baboon system, we have evidence for parts of this causal chain. For example, we have evidence for a link between early life adversity and fitness, whereby individuals who experience more adversities during their early life have reduced lifespan. We also have evidence for the relationship between early life and adult traits, as shown by the orange arrow, where early life adversities decrease female-female social bonds, increase or have no effect on female-male social bonds, and increase glucocorticoids. In addition, there are links between adult traits and fitness, as shown by this purple arrow, where decreased social bonds and increased glucocorticoids are both related to decreases in lifespan. Therefore, while it's clear there are links between early life adversity, these adult phenotypes, and fitness, it's unclear if the effects of early life adversity on survival are mediated by social bonds and or glucocorticoids. So in other words, we don't know if early life adversity affects social bonds and GCs, which in turn affect fitness, or if there are direct effects of early life adversity that are not mediated by these adult traits. So today I'm going to tell you about a study where I've been able to link real-time data on early life adversities to observations of social relationship, endocrine data, and survival in the same individuals. And in doing so, we also provide a test of the biological embedding hypothesis, which predicts that animals who experience early life adversity will have weaker social connections, elevated glucocorticoid levels, and shorter lifespans than individuals who do not experience poor early life environments. In addition, this hypothesis predicts that part of the effect of early life adversity on lifespan will be mediated either by social connections and or glucocorticoids. To test our hypotheses, we took advantage of a data set from the Amboseli Baboon Research Project, which studies wild baboons located in southern Kenya and northern Tanzania. This project is led by Susan Alberts, Jenny Tung, Beth Archie, and Jean Altman. And data has been collected on 2,000 individual wild baboons living in free-ranging social groups continuously since 1971. And these data are collected by our experienced observers, Kinua, Raphael, Langita, and Sarah, who have been with the project for decades or longer. And none of this data would be available without these wonderful observers and our support staff who are pictured here. Amboseli baboons are part of an admixed population of wild yellow and Anubis baboons who live in social groups of 20 to 150 individuals. Males disperse around maturation, which is about 7 to 8 years old for males and 4 to 5 for females, where females remain in their maternal social group throughout their life. And for females, which are the focus of this study, they tend to live on a median lifespan of about 18 years. From the MSLA Baboon Research Project, we compiled a data set of 199 females. And from these 199 females, we have 1,849 years of life, where we measure social relationship and as well as survival data. We have almost 12,000 glucocorticoid samples, and we've recorded 74 deaths. Here, we are only modeling female for survival because, as I said, males disperse from their natal groups, which makes estimating survival for males a bit more complicated. Our measure of early adversity was this cumulative adversity index, which has been shown to lead to reduced lifespan in previous studies of this population. And the cumulative adversity index is the sum of 6 sources of early adversity. 2 of these early adversities have to do with reduced resources. So if an individual experiences drought in the first year of life, or if they have a high group size at birth. 2 of these adversities have to do with the maternal social environment. So if the individual's mom was of low dominance rank when they were born, or had low social connections. And 2 have to do with family structure. If an individual's mother died before she reached 4 years of age, or if the individual had a close in age younger sibling who might provide competition over maternal resources. One of our measures of adult phenotypes was social bond strength, which we measure by the dyadic sociality index, which we calculate on a yearly basis for every individual female. And this is a measure of social bond strength of the top 3 social partners a female has during that year. And it's based on grooming relationships, which are a common affiliative behavior in baboons and other primates. Negative values indicate weaker social bonds than the population average, whereas higher values indicate an individual had stronger social bonds on average. We also use glucocorticoids, which are a steroid hormone related to the maintenance of homeostasis and ultimately related to the stress response. Here we're using fecal glucocorticoids, which are integrated over hours to days and therefore represent a longer term measure of an individual's endocrine profile. As I said, we had almost 12,000 glucocorticoid samples, and for each individual, we had a mean of almost 60 samples taken over 6 years of their adult lives. And I'd like to take a moment to thank Laurence, who is our hormone lab manager, without which these data would not be available to us. So to test our hypothesis, we are using mediation analysis with joint modeling. And this approach is being developed by 2 of our collaborators at Duke, Dr. Fan Li and Shushi Zhang. And this approach allows us to take into account the longitudinal nature of our mediators to examine if they're the black box mechanism that links early life adversity and survival. We hypothesized that social bonds with females or social bonds with males or social bonds with or glucocorticoid levels might actually mediate the relationship between adversity and survival. And this framework allows us to test if social bonds or glucocorticoids are part of this chain that link early adversity and survival. And so in other words, when I say that we're testing a mediation effect with mediation analysis, I mean that part of the reason that early life adversity affects survival is because it affects social bonds or glucocorticoids. Mediation analysis also allows us to estimate parts of this framework. So for example, we can test this orange arrow, which is the effects of cumulative adversity on social bonds. And we can determine the effects of social bonds or glucocorticoids on survival independent of changes in cumulative adversity levels. This framework also allows us to estimate the total effect of adversity on lifespan, and this can be broken down into 2 parts. One is the direct effect shown in green, which is the effect of early adversity on survival independent of changing in the mediator, so independent of changes in social bonds or glucocorticoids. And secondly, shown in pink, we can estimate the indirect effect, which tells us how much of the effect of early adversity on survival is due to early adversity changing social bonds or glucocorticoids, which in turn alter survival. So first, we found that for each additional source of early adversity a female experiences during the juvenile period, her lifespan is decreased on total of about 1.5 years. And while we don't see a significant direct effect of cumulative adversity independent of the effects of social bonds, the magnitude of the direct effect is almost the same as the total effect. For each additional source of early adversity a female experiences during her juvenile life, her social bond strength is decreased. And for every 1 unit increase in social bond strength, her lifespan is increased by 2.2 years. So social bonds increase survival. In addition, we have a significant mediation effect, which means that for each additional source of early adversity, 1 month of the difference in lifespan for females that experience early adversity is explained by the pathway through social bonds. So in other words, early adversity decreases female-female social bonds, which in turn decrease survival. However, this causal pathway through female DSI only explains about 5% of the total effect of early life adversity on survival. And in addition, if a female experiences all 6 cumulative adversities, her lifespan is reduced by only 6 months, which is not through social bonds, it's only 6 months. Okay, so in addition, for male social bonds, we see that the effects are similar magnitude, but in the opposite direction. So for the pathway through female-male social bonds. So for each additional source of early adversity, a female's bonds with males are actually increased rather than decreased. And again, increased social bonds increase survival. We do again have a significant mediation effect, but it's positive rather than negative, meaning that the pathway through female-male social bonds may counteract the negative effects of early life adversity. So in other words, early life adversity increases female-male bonds, which in turn increase survival. And this causal pathway through female DSI, again, explains a little bit more than the female-female bonds, but explains only about 9% of the total effect of early adversity on survival. For glucocorticoids, we see that they do not mediate the effects of early life adversity on survival. For each additional source of early adversity, we do have a strong effect on glucocorticoid levels, whereby individuals who experience more adversities during their early life or have higher glucocorticoid levels in adulthood. In addition, higher glucocorticoids in adulthood decrease survival. However, although it's of similar magnitude to the social effects, we do not see a mediation effect of GCs, which in part may be driven by the higher variance in glucocorticoids. And this means that there is not a pathway through GCs that it can explain the effects of early adversity on lifespan, and suggests that early adversity in glucocorticoids might have independent effects. To sum up this part of the talk, we see that females who experience early life adversity have weaker social connections than those who do not. However, in contrast to the predictions of the biological embedding hypothesis, they have stronger social connections with males. Females who experience early life adversity also have elevated glucocorticoids and shorter lifespans. And while part of the effect of early life adversity was mediated by social connections, we saw potentially independent effects of glucocorticoids. And so in whole, I'm providing some mixed support for the biological embedding hypothesis in the baboons. However, in all cases, the mediation effect was quite weak, despite somewhat strong effects of these individual pathways. So we wanted to understand if this had to do with a true weak and small mediation effect, or if there could be some underlying data structure that we're missing with these analyses. So for example, I showed you that for female-female bonds as a mediator, we see strong effects of early adversity on survival, coupled with effects of early adversity on female-female bonds, and effects of female-female bonds on survival. But again, very weak indirect effects. And so we hypothesized that this weak indirect effect might be due to time variation in how early life adversity affects mediators and how mediators affect survival. So in order to address this, we built a simulation model. And in this model, early adversity can affect the mediator, which in this case, we're using female-female bonds as an example. And this effect on the mediator can occur primarily early in adulthood or primarily late in adulthood. And of course, bonds in early adulthood can affect survival early in adulthood, but for now, we're going to assume that all individuals that will survive this early time step, and we model survival later in adulthood. Survival later in life could, of course, be affected by social bonds later in life, or it could be more affected by social bonds in the past, so social bonds early in life. And we use this model to generate predictions about how timing effects of early adversity on the mediator and the mediator's effects on survival could influence the size of the indirect effect. And so we find that the timing of these effects is key to the size of the indirect effect. Specifically, if the timing is matched, we expect to see a larger indirect effect than if the timing is mismatched. So for example, if the effect of early life adversity on female-female bonds is greatest early in adulthood, and social bonds early in adulthood predict survival at this later time, then we expect to have an indirect effect that's intermediate or large in size. Similarly, if the effect of early adversity on female-female social bonds is greatest later in life, and survival later in life is affected most by bonds late in adulthood, then we expect a large indirect effect. However, if the timing is mismatched, indirect effects are likely to be small. For example, if early adversity affects bonds early in life, but survival is dependent on bonds later in life, then we see a small indirect effect. And similarly, if early adversity affects bonds late in adulthood, but survival is dependent on bonds early in adulthood, then we expect a small indirect effect. And so this model allowed us to generate predictions as to what could be driving our small indirect effect in the baboons. So next, I'm going to show you data on how the baboon system varies in the effects of early adversity on the mediator and the mediator on survival. And so first, I'm going to show you how the effect of early adversity on female-female bond strength varies with the age of the focal female. On the x-axis is the age of the female, and on the y is the effect size of the relationship between early adversity and the mediator. And what we see is that as females age, the effect size gets more strongly negative, which means that early adversity weakens female-female social bonds, but this effect is strongest later in a female's life. And so this suggests that our results are falling somewhere in the right-hand column. However, dependent on how survival is related to mediator values, this could be a small or a large indirect effect. So we estimated that in the model. And so here the x-axis is the effect size of the current mediator, or the effect size of the current mediator in survival. So this is here, which is the early effect in our or the late effect in our model, versus the effect size of the past mediator on survival, which is the early effect in our model. And the y-axis here is hazard ratio. So more negative numbers means that increased female-female bond strength increases survival. And we see that current female-female social bonds have almost no effect on survival. Instead, female-female social bonds in the past strongly affect survival. So here we're showing the previous year's social bonds as the past measure, meaning that if social bonds for a female with other females were strong in the previous year, she was more likely to survive than if they were weak in the previous year, or stronger weak in the current year. And so in the Amboseli baboons, the effect of early life adversity on the mediator are strongest later in life, but survival is dependent on the mediator values early in life. So under these conditions, we expect that we see a small indirect effect, which is what we see. So to sum up this part of the talk, it's likely that our small indirect effect is due to mismatched timing. We see the effects of early adversity on female-female bonds are stronger later in life, while survival depends on female-female bonds earlier in life. And so under these conditions, we expect to see weak indirect effects, which is what we see. And so as a whole, I've showed you mixed support for the biological embedding hypothesis in the baboons. While social bonds with females and males mediate the link between early adversity and reduced lifespan, social bonds with males act as a counteracting force to the negative effects of early adversity, and they may act as a source of resilience to early adversity, which is something that we're following up on. I've also showed you independent effects of glucocorticoids and potentially early adversity on survival, suggesting that by considering this whole pathway, we can gain insights that were not obvious from focusing on parts of the pathway. So from before, we saw strong effects on early adversity on glucocorticoids and of glucocorticoids on survival. So we hypothesized that these would be linked, but we did not see that. In addition, we hypothesized that the timing of these effects is key to assessing indirect effects. And therefore, by considering how effects vary across the life course, is really important in understanding how these relationships between juvenile phenotypes, adult traits, and fitness are related. And finally, I just want to say we're working, and by we, our statistician collaborators mainly are working on developing mediation models that include time varying and time-length effects. So I'm really excited to talk about that at potentially a future meeting. And so with that, I'd like to thank everyone involved with the Amboseli Baboon Research Project, especially our observers, and as well as the Alberts Lab and my funding sources. And with that, I'm happy to take any questions.
All right, if you have any questions for Liz, please drop them in the chat or in the Slack. While we wait for people to type, I'm not seeing any yet. I have a quick question. So you lumped all the adversities together in your model, and I'm wondering if different adversities could have different effects. So for example, maybe drought has bigger effects on court, or maternal loss or maternal low rank could have a different effect on social bonds. Any thoughts on what would happen if you kind of broke them down a bit?
Oh yeah, so we also do break them down. It's just a lot for just a 20-minute talk. So we see that on a whole, maternal loss has one of the strongest effects on lifespan as well as these mediators. And we also see, as you mentioned, that drought has a pretty strong effect on both glucocorticoid levels and social bonds. And what we're doing as a next step is trying to understand if any of these effects could interact with each other. Potentially maternal loss is an amplifying effect, and so it's really bad to have maternal loss in concert with something else rather than just two other adversities. So stay tuned for that as well. Good question.
We have one more quick question before I move to the next talk. I probably missed this, but what affects social bonds early in life?
So I didn't actually look at social bonds early in life, but the social bonds of the mother early in life affect social bonds in adulthood. So that's a good question. And we do have a collaborator looking at how adversities and other circumstances early in life affect social bonds in the juvenile period as well. So I'll let him talk to that. Thank you.
Perfect. Okay, so on to our next talk. We have Alexandra Hooks, who will be telling us about polyandry in a marine gastropod. Looks great.
Sorry, I did that in the wrong order of opening and closing things. So today I'll be talking about some of my dissertation work, which is the evolution of polyandry in a marine gastropod. So multiple mating or female multiple mating, known as polyandry, is common in many different taxa and is ubiquitous across the animal kingdom, including in vertebrates and invertebrates. And it's common both in terrestrial environments and in the marine environment. And so there are several hypotheses to why this mating strategy may have evolved in these different systems. And so just to quickly go over those hypotheses, they can include direct benefits to female fitness through increasing mates, increasing nutrition via nuptial gifts. This is common in a lot of insect species during mating. Increasing female stimulation, so more eggs are produced. Increased in parental care and the replenishment of sperm. Females may also mate multiply to find good males that increase the number and survival of their offspring, and this can be done about two ways. They could be mating multiply to find the best male in the population that would create the most fit offspring, which is considered the good genes hypothesis, or they may be looking for the most genetically compatible male for themselves, or as a way to prevent mating with only incompatible males. And then lastly, the last hypothesis I'm going to discuss is the females mating multiply to produce genetically diverse offspring to increase their offspring survival. They can do this by genetic diversity, increasing the likelihood that some offspring will have the viable genes they need in a particular environment. And genetic diversity can increase the range of offspring phenotypes, which can increase the likelihood of some of their offspring surviving. And so you see this, this sort of bet hedging in heterogeneous environments where the mother can't predict what would be best for the environment the offspring will experience. And so a lot of these hypotheses that I mentioned have come from a limited amount of taxa and life history types. For instance, a lot of what we know comes from insects, birds, and frogs. And in each of these cases, many of these systems can have complex social behaviors, including mate choice and high parental care. For the marine taxa, these types of questions are almost exclusively from broadcast spawners, where multiple paternity is almost inevitable, and studies have come from mainly sea urchins and fish. And so what we don't know is which of these hypotheses are translatable to a wider spread of taxa and life history types. And what I mean by that, I'm including systems that have no to little social structure, no parental care after laying, no obvious pre-copulatory mate choice or mating displays. We also need more experimental studies on wild populations, as a lot of studies, especially in the insects, have been on lab population. And we also know very little about marine copulatory systems, with only a few papers directly testing these hypotheses, and compared to the terrestrial environment, we know almost nothing. And so, therefore, my dissertation has focused on addressing this gap by testing multiple hypotheses for polyandry on an easily manipulated marine gastropod that exhibit these characteristics, and these characteristics can be pretty common in marine systems. And so the system I use is the Florida crown conch, as shown here, where the snails are, they're carnivorous and scavengers, eating both bivalves and mollusks. They have separate sexes, so they're copulating species with no parental care, no obvious mating display, and little social structure. Both males and females mate multiply, so 5 to 7 years. And they can store sperm during their breeding season, which lasts about 4 months, but they don't overwinter their sperm. And females lay egg strings with multiple egg capsules as shown here. So what you're seeing here is that a female is mating with a male, and then she's also laying these egg capsules. And inside of each of these egg capsules, so these egg capsules are about the size of a penny, and she can have around 50 to 150 offspring in them. And she'll lay around 8 to 18 of these egg capsules at one time on an egg string. And inside of these egg capsules, with these 50 to 150 of embryos, they usually occur with multiple paternities inside of them. And so with this system, I wanted to ask the questions, do more mates equal more offspring? Does multiple mating increase phenotypic variation within broods? And do more mates, are they equal to the proportion number of sires? And so I did this by mating males and females in the lab with only one mating pair in a tank at a time. And so here you can actually see the copulating of this species. You see the male here, his penis is interjected into the female. And so in the lab, you can get confirmation of mating, of confirmed matings. And so after the pair have separated, the male was removed and either another male was introduced to the female or not, depending on her mating treatment. And so females would lay their egg strings, and those egg strings were placed into a sterile lab tank until hatching, as shown here. And I should note that females can lay multiple egg strings during their reproductive season, but I'm only going to show you data from their very first egg string in which they laid. And I'm now wondering, are my slides cut off, or can everyone see that?
Looks good to me.
Okay, perfect, thanks. So I carried out these mating experiments for 2 years with both unique individuals in each year and unique males for every mating. Females were isolated for several months to over a year, to several months in 2019 and over a year in 2018 to account for sperm storage. In 2008, that long isolation in 2018 is what helped us know how long they can store the sperm. And so in 2018, I mated 28 females with a range of 0 to 5 fathers, 5 males. In 2019, I upped the ante and mated 53 females with either 1 or 4 males. And so data collected during these mating experiments include hatching and egg capsule number in both years, and hatching size and their corresponding DNA for 2019 only. And so for the first question of, do more mates equal more offspring? I'm looking at both years for this data set. I wanted to look at the total number of offspring per mother, if females laid egg capsules or not, so their probability to lay, average number of hatchlings per egg capsule, and then the number of egg capsules that a female laid. And so what I found that the number of mates increased the number of offspring only sometimes. In 2018, the total number of hatchlings per mother in the 1X treatment was very similar to those that had been mated with multiple males. However, in 2019, 1X females lay significantly less offspring than females that had mated multiply. And so to look at this pattern a little bit more in detail, I started looking at the probability to lay in each treatment by year. And so what we found was that in 2018, every female laid regardless of her treatment, while in 2019, we found that 1X females were a lot less likely to lay than multiple mated females. And so I want to also note that in this 2019 data set, for the females that laid egg strings, females were not producing more egg capsules, nor having more hatchlings within an egg capsule. And so the difference in the offspring total that I've shown in the previous graph is solely driven by this probability delay that I'm showing you here. And so I wanted to look at factors that could be potentially driving the differences in probability delay between years and treatments. And because I'm doing this post experiment, it's looking at any sort of patterns or trends that came out in the data. One trend that I did find in my data is that total copulation increased the probability of females to lay. And so looking at 1X mothers only, in the blue lines here, you have 2018 data where every female laid. And you can also see that in 2018, they had higher copulation times than the majority of 2019 females, which are in red. And also in 2019, you can see that on average, females with higher copulation times were more likely to lay than not. And so when I looked at the multiple mated treatments, for mothers that mated multiply, we can see a flattening of this curve. And this is somewhat expected since the majority of females laid, and this could possibly be due to the high copulation time in the MX treatment. Because as you can see here, no female mated more than 480 minutes in the 1X treatment, while almost all of the females had mated over 500 minutes in the MX treatment. And so to sum this up, do more mates equal more offsprings? The trends we have found is that increasing the probability delay was directly affected by increasing the number of mates, and this could be affected by the increase in copulation time. And so what this is suggesting in the system is that multiple mating may have evolved for the direct benefit of sperm supply. And so for my second question was, does multiple mating increase phenotypic variation within broods? Because these are intertidal species, they exist in a very heterogeneous environment. And so it may be beneficial for females to produce offspring with high phenotypic variation in the hopes that some of the offspring phenotypes will survive. And so to investigate this question, I compared offspring size and variation of offspring size in a brood to see if that was happening. And so I actually found that the mean offspring size per mother did not differ between treatments as shown here. And they actually are very similar in size of around 815 microns, almost to the dot. And that also the coefficient of variance in offspring did not differ between treatments as well. Just so you know the numbers, the mothers behind this, but so you know the number of hatchlings behind this. I measured around over 5,000 hatchlings in both of these for a total between these two treatments. And so to sum that up, does multiple mating increase phenotypic variation within broods? The answer is no. Number of mates did not increase offspring size, nor did it increase the coefficient of variance of offspring size, so variation within a brood. And so what this is suggesting is that multiple mating did not evolve to increase the phenotypic variation, which we might expect in this system. And then my last question was, do more mates, are they equal to the proportional number of sires? So are males contributing equally to a brood, or are certain fathers dominating this brood? This is important because if females were mating to increase genetic diversity of their offspring and hope that some of their offspring will have the genes most suited for that environment, then you might expect the brood to have many fathers and for those fathers to be contributing equally. And so to investigate for the multiple mating treatment, I did a parentage assignment of offspring with their known parents. And since this is from the 2019 data only, all females were mated with 4 males in this treatment. However, what I found was that the number of sires at hatching was only around 2.7, and there was variation in the number of sires within a brood, even though all the females were mated with 4 males. And so from this, we know that not all matings result in offspring being produced. And so not only with the number of sires, I also wanted to know if the sires were contributing equally to a brood. And so to look at this, I investigated the effective number of sires within a brood. So to explain effective number of sires, I'll give the example of if you have 4 sires contributing equally to a brood, then the effective number of sires would be equal to 4, be equal to the number of sires they had. If instead you had 2 sires, where one contributed to 80% of the offspring in the brood, while the other father only contributed to 20% of the offspring, your effective number of sires would be around 1.4, which is less than the total number of sires. And so I did this with my own data and found that the effective number of sires actually equaled 1.7, which was less than the number of sires and less than the number of mates. And so what this is telling us is that 1 to 2 males tend to dominate a brood, and that the genetic diversity of offspring is not as high as you would expect if females were trying to produce a range of genetically diverse offspring. So on a side note, I wanted to know why certain males were dominating a brood, why we have this pattern. And so preliminary data so far is showing that mate order can play an important role in this, where the first 2 males are contributing a higher proportion of offspring than the subsequent than the rest of the males, and that the probability of them siring any offspring decreases as their mate order goes further down. And so other factors that I'm actively analyzing is the effect of male copulation time on whether or not how much they sired, male size. And I'm also will be comparing shifts in paternity from embryo to hatchling stage to account for sire-based mortality during development to see if certain fathers had a higher likelihood of surviving during development. And so for this third question of do more mates equal a proportional number of sires, the answer is no. 1 to 2 males dominate an egg capsule or egg string. And so what this is suggesting is that multiple mating did not evolve as a way of risk spreading, which is supported by the lack of variation in the phenotypes within a brood, and by the fact that the offspring were not very genetically diverse, as only 1 to 2 males dominated a brood. And so the take-home messages for this work is that multiple mating provided direct benefits to mothers by increasing the total number of offspring produced. Multiple mating likely evolved for sperm assurance, as total copulation time and number of mates increased the mother's probability to lay. Multiple mating did not evolve, or at least it's suggesting did not evolve as a way for risk spreading, as offspring were not very diverse both genetically or phenotypically within a brood. However, for this particular point, we can't discount that maybe selection is favoring the mothers to have this genetic bet hedging or increase in genetic diversity, but that selection is being overwhelmed by the selection on males to find ways to dominate the brood. And also the multiple mating to prevent male harm is unlikely in the system, as females can mate minutes after mating with another male, and she can reject most males when mating is unwanted. And so for that, I would like to acknowledge my funding sources, my field assistants, and I will take any questions.
Great. Thank you so much for the fantastic talk. We have our first question here. Does number of offspring increase proportionally with effective mate number? Effective mate number increases 50%. Does offspring number increase 50% as well?
That's a good question. The answer to that is no, because even in 2018, when I had a range of 2 males to 5 males, they still had a very similar mean number of offspring. The mothers did. So it felt like more of an all or nothing. She either had enough sperm and then she laid, or she did not have enough sperm and didn't lay. And the number of mates didn't really affect how much she was laying. It was more of, did she have enough sperm to lay those eggs? At least that's what I think.
Great. People can type in more questions in the chat. Meanwhile, I'll ask you quickly. I was wondering, well, you said that you're going to look at the relationship between duration of mating and proportion of offspring, which I think seems from your first results to be really relevant. I guess related to that, if you do find a relationship, what determines the duration of the mating? Who stops a mating bout? Or what is the kind of mechanism that determines how long a mating bout is?
So what I've noticed for watching these snails for more years than I would like, the female will actually start, in most cases than not, the female will disengage and start and move away from the male. And so she can reject males and stop the males from mating. The only time that can't happen, or she chooses not to do that, is when she's laying. So when she's laying, she can have the choice of aborting those eggs and moving on, or just mating. And so that's the one case in which she will just continue to mate. But if she's not laying, she can move away and disengage.
Awesome. Thanks. I think it's time for the next talk.
Yep, next up we have Brian Lurch, who's going to tell us about same-sex sexual behavior and indiscriminate mating strategies.
Well, thank you all for coming. There we go. So I'm going to start off actually with a question, which is, what do all of the organisms on this slide have in common? And in particular, what about their behavior can we think of that they all have in common? And if you hadn't just heard what the title of my talk was going to be, something I would bet most of you wouldn't guess is that same-sex sexual behavior, or as I'll refer to it, SSB, has been observed in all of those different species. In fact, it's been observed in well over 1,000 species, and clearly based on that last slide, that's in a really wide range of taxa. So I'm going to be telling you about one potential explanation for why SSB is so widespread, and that's that it results from indiscriminate mating. This is work that I've been doing at UNC with Maria Servideo. So historically, researchers who have noticed animals engaging in SSB sort of tend to point out the same things. They're quick to point out that they expect this behavior to come with costs. It's repeatedly referred to as an enigma, a paradox. And in spite of this, there's actually been a wide range of explanations for why animals engage in SSB. I'm going to point out before I walk you through these that most of these don't imply anything about sexual attraction. In particular, what I'll be talking about sort of does not include a notion of sexual attraction. Definitely does not apply to humans, so I just want to sort of make that clear up front so people don't sort of go there. So one explanation is that it results from different types of non-Mendelian inheritance. There are some potential social benefits to SSB. This is most often probably referred to in bonobos, where it's known that sex is used as a means for reducing conflict. It could be sort of an extension of male-male competition, allowing for a sort of manifestation of aggression against rivals. There's some evidence that SSB results as a byproduct of high sexual drive. It may even provide a means for extracopulatory insemination. There's some evidence in flower beetles that a male will mate with another male, and that second male can actually transfer the first male's sperm onto females. And like I said, the explanation that I'll be talking about today is that it results from indiscriminate mating. So what do I mean by indiscriminate mating or indiscriminate sexual behavior? Is just that animals could engage in matings without first attempting some sort of sex-based mate identification. The result of this would be that animals are engaging in both same and opposite or different sex matings. This was hypothesized recently, just over a year ago, to be the ancestral mode of sexual behavior. Although a response to that paper basically argued that, well, even if it's ancestral, we shouldn't expect it to stick around, basically repeating some of those arguments on the first slide that you could easily imagine that the indiscriminate mating would quickly evolve away because SSB can't have any benefits. So what this tells us is that potentially, for us to think about how important indiscriminate mating is, we need some explanations perhaps for how it could persist through evolutionary time. And I'm going to argue that there's kind of two features that have not received enough attention in the literature about animal reproduction that could potentially explain why indiscriminate mating is persisting through evolutionary time. The first one is just that perhaps animals are not able to accurately determine the sex of their conspecifics. I'd imagine that a number of people on this call work with organisms that are challenging to sex, and perhaps the same is true for individuals of those organisms, that sort of signals or cues that allow them to determine whether a potential partner is of the same or opposite sex is difficult or even impossible to detect. And the second is this idea of search cost. Now, this has received some attention in the context not of SSB, but in sexual selection and mate choice. And perhaps there are search costs that result from this increased choosiness that indiscriminate mating is able to avoid. Maybe attempting sexual discrimination, or attempting to discriminate the sex of your partner, kind of carries with it some sort of additional cost, such as a predation cost. So I'm going to develop a mathematical model to sort of test some of these different ideas. And this model has two sexes. The first one I'll refer to as the searching sex. This is the sex that seeks out potential mates and could potentially discriminate the two sexes. The second sex being the targeted sex, and they are capable of providing some signal or cue of their sexual identity. So the searching sex can attempt to mate preferentially with the targeted sex. This is what I'll refer to as discriminant mating. And we see that in this individual in the top right that's directing more matings to the targeted sex than other members of the searching sex. We'll assume that this carries some cost to survival, this being the search cost I talked about on the previous slide. Conversely, members of the searching sex could mate indiscriminately, like this individual in the bottom left that's directing matings equally at members of both the searching and the targeted sex. So then the targeted sex, like I said, is capable of displaying some signal that basically tells others that they are of the targeted sex, such as this individual here. And the presence of, the absence of this signal means that individuals that are mating discriminately will not preferentially mate with individuals of the targeted sex that aren't displaying that signal. So just to sort of formalize this a little bit more, we'll assume that the searching sex, this sex that's capable of discrimination, mates exactly once per reproductive bout. If they are able to successfully discriminate a partner of the opposite sex, they'll do so. Otherwise, they'll mate indiscriminately. And the probability that the searching sex mates as a result of discrimination, so only with the opposite sex, is given by this product AS. Then we see what's graphed here, red being the highest probability there for matings resulting from discrimination. And like I said before, it takes two steps, right? There's the attempt to discriminate, which has to be high, and there's also a signal or cue from the targeted sex that has to be picked up on. So even if there's the attempt to discriminate, if there's no signal that's detectable, then we'll see that members of the searching sex will have to mate indiscriminately. And stronger discrimination attempts, so moving to the right on this graph, means lower survival probability as a result of that search cost. So we can use those assumptions. You don't have to worry about the specifics of the slide. You can use those assumptions to write down an equation for expected lifetime reproductive success. And we can basically optimize that equation and ask if indiscriminate mating is ever the optimal evolutionary strategy. And I should say here that we also built a corresponding population genetic model that's a little more explicit in some of the details of how these things work out, not just an optimization procedure. And the results between those two models are basically identical. So from henceforth, I'll just be talking about this optimization procedure. Okay, so what are the optimal discrimination strategies? So we can look here. This is the case of high mortality. X-axis is the cost of discriminating, again, that search cost. The signal from the targeted sex is the y-axis. And we see that over a wide range of these parameters, indiscriminate mating in black is the optimal strategy. And we also see sometimes complete discrimination evolving. This is unsurprisingly most likely to happen with low search costs or when that signal from the targeted sex is strong. All of these gray regions would be imperfect discrimination evolving as the optimal strategy. And we see that across parameters in this model, we have some region of parameter space where indiscriminate mating is the optimal strategy. Interestingly, it's the least common at intermediate mortality rates, this middle graph. And the way you can understand this intuitively is that at low mortalities, individuals are likely to have a large number of mating attempts in their lifetime. So a single round of SSB is unlikely to have large fitness consequences because they'll again have many mating attempts later in life. Whereas at high mortality, it's sort of the opposite going on. Individuals are unlikely to ever get to mate, and what this means is basically they're just trying to survive to get to mate once. And then they can rely, the way to maximize the probability they get to mate is to mate indiscriminately. And then sort of to get a fitness benefit from that, they're basically just relying on chance. The important thing to note about how to interpret these results is it means that same-sex matings will occur as a part of the optimal evolutionary strategy. So black in these graphs now is basically around 50% same-sex matings given the optimal strategy, and we see that only when the search costs are really low and the signal is very strong do we see basically no SSB. So the majority of parameter space does show some SSB as the optimal strategy. But we can also imagine that sexual signals are also able to co-evolve with this discrimination attempt, and it's sort of easy to imagine that these two traits can become genetically associated and sort of drive each other up in frequency. So the question then becomes, can indiscriminate mating still ever be the evolutionary endpoint when these sexual signals are also evolving freely? So we're going to look at results from this co-evolutionary model a little bit differently. So this graph is going to be initial frequency of discriminators on the x-axis. So 1 is all of the searching sex in the population is attempting to discriminate. 0 is none of them are attempting to discriminate. This is initial frequency signaler. So down here on the y-axis, we have none of the members of the targeted sex are providing the signal. Up here, all of them are providing the signal, and we get 2 regimes. So when there's high enough initial frequencies of both discriminators and signalers, we get complete discrimination and perfect signals evolving. Whereas if we have either low initial frequency of discriminators or sufficiently low initial frequency of signalers, we get indiscriminate mating and no signals evolving. So we get this biastability, depending on initial conditions, we can fall into either of these 2 regimes. So if we zoom out and look at these graphs across a wider range of parameters, so across mortality rates on the sort of moving across the top, and moving down the columns, higher costs to both discrimination and signaling, we see the picture is almost always the same. Most of these graphs have this biastability where, if we have low enough initial frequency of signalers and discriminators, then no signaling and indiscriminate mating will evolve. But if we have sufficiently high frequencies, then conversely, we'll go to this regime where we have both discrimination and signals evolving. What's important, though, is close to 0, 0, we're always in the green region. You can actually prove this analytically. So what this tells us is that, if we're sort of starting at this condition where there are no sexual signals and there is indiscriminate mating, then we should expect evolution to sort of be trapped there. It would always stay at this place where there's no signals and discrimination does not evolve. So this seems to suggest that if discrimination were to evolve, then probably either discrimination itself or those sexual signals must be ancestral. I tend to think that, again, that paper I pointed out earlier that argues that indiscriminate mating is likely ancestral, I tend to agree with that. I think that what these results kind of suggest is that there are some detectable differences in cases where we see discriminate mating evolving. There probably are sort of as a byproduct of the evolution of 2 sexes, detectable differences between those 2 sexes. that individuals can pick up on. So in conclusion, I've shown that indiscriminate mating can often be an optimal evolutionary strategy. And perhaps this can explain SSB in some taxa. And I think in terms of thinking about where to take this for the future, the presence of these readily detectable differences between the sexes hasn't been given sufficient consideration in terms of thinking about trying to measure these things or figure out really what it is that individuals are detecting when they're trying to differentiate between two sexes if they are. Yeah, so that's all I have. Thank you for listening, and I'm happy to take any questions.
Great, thank you for the wonderful talk. We have one question coming in already. Super cool talk. How realistic do you think the range of costs of discrimination shown in your figures is representative of what happens in nature? For example, does the cost of 0.05, what does the cost of 0.05 mean biologically?
Yeah, that's a really good question. So the cost of 0.05 would mean that there's basically a 5% reduction in survival probability as a result of attempting to discriminate. Is that realistic? I have no idea. And people just really haven't tried to measure search costs in the context of SSB. And I would love to see that. Yeah, but at this point, I think we really just don't know what those search costs would look like if they even exist. Yeah.
Another question here. Is there a possible role for antagonistic behavior from same-sex individuals?
Is there a possible role for could you maybe elaborate on that a little bit?
So I have Stephen, do you want to elaborate a little bit, either by unmuting or taking a bit more? So I'll guess here, I guess aggression from same-sex individuals, will that influence your results?
Oh.
Like if same-sex individuals can identify each other and then be aggressive.
Yeah, so I actually hadn't thought about that. Yeah, what would that do? I mean, I could speculate wildly that, perhaps that could make it more favorable to attempt to discriminate, because then you would have both, yeah, because then you could have both sort of the benefit of being able to identify mates and the ability to sort of, antagonize your competitors, which seems like it could make discrimination more favorable. But yeah, honestly, I hadn't thought about that. So that is just wild speculation at this point.
Here's another one. Thanks so much for the great and thought-provoking talk. I'm curious if you've considered or plan on considering the role of varying sex ratios. It seems like that could affect the results, but may not have costs or have different costs.
Yeah, so we actually already thought about, it's one of those things we already did, but to fit it into 15 minutes, I didn't really talk about. So we see that when the sex ratio is skewed in favor of the searching sex, so there's more searching sex than targeted sex, is also another condition that would make discrimination more favorable. So you're most likely to get indiscriminate mating when there's more of the targeted sex than the searching sex.
Here's another question that came in. Given that when SSB is detected in nature, it is usually a form of imperfect discrimination, yet the model indicates the most likely outcome is either absence of or perfect sex discrimination. Does this model support mistaken identity or lack of discrimination as an explanation of observed SSB patterns?
Yeah, so I think this sort of goes back on in terms of would we expect to see, do we see less, is imperfect discrimination more common in nature than in the model? I think we just don't know because, getting back to that first question, we don't know what the reasonable parameters are in order to ask that question. Does it support, so I think in terms of the thinking about like the mistaken identity hypothesis that that you're referring to, I think it sort of shines an evolutionary light on the mistaken identity hypothesis in the sense that it's saying, okay, maybe it's not just that there's some constraints that are, the individuals could be doing even better if they were engaging in less SSB. It's sort of saying, oh, well, maybe if we think about these costs and we think about the signals they're detecting, maybe, maybe it's not necessarily that they're making more mistakes. It's that, this is sort of the best that they could be doing.
One last quick question, and then we'll move on to the next one. Jeremy is asking about the whole alpha equals 0 line. Is it stable to invasion?
No, not if you have sufficiently strong signals that you can invade off of that line.
And just privately, I got a message from the person who asked the previous question saying that I would argue that it's the same hypothesis. I think there might be some discussion on Slack that is going on, and one more question that if you want to answer offline in the chat, that would be great. We need to move on to the next. Thank you so much.
Okay, great. Our next talk is Hector Douglas, who will be telling us about a novel bill pigment.
Okay, can everyone hear me okay?
Yep.
Okay, good. So this picture is showing a male there on the left and a female crested auklet, both displaying these brilliant orange accessory bill plates during the breeding season. In my talk, I'll be discussing the phenology of pigment expression, the apparent disruption of trait expression by a marine heat wave, a potential link of the trait to diet and ecology, and links to between the pigment behavior and physiology. So this is showing the pigment molecules. This is a cross-section of a crested auklet's accessory bill plates. The pigment molecules are secreted into the keratinized layers, and the microscope slides are shown here, the cross-sections, and the slide on the right, higher magnification, you can see the boundary between epidermal and dermal layers evident there. So this pigment is acquired during the month of April based on a phenology study that I did with the Cincinnati Zoo in 2006. This composite image shows various individuals in their captive bird collection at that time, recorded on April 4th. By the end of April, the beginning of May, all of these birds had completed acquisition of this orange bill pigment. During the summers of 2015, 2016, I conducted research on crested auklets at Little Diomede Island, which lies in the middle of Bering Strait within a few miles of the international date line in Russian territory. A broad and shallow ocean shelf spans Bering Strait, and you can see this green highlighting there, which signifies an extension of the Bering Sea greenbelt. It's a zone of very high productivity that's advected across the shelf, and the auklets are dependent upon that food web. So you see the crested auklets here over in the top right panel. They occur in very large colonies, and they forage in the waters adjacent their breeding colonies. So they have a relatively limited range, and they integrate the their diets in their tissues. And they forage relatively low in the food web, close to the base of the food web. So I used spectroscopy and other methods to study pigmentation. Figure on the right shows some work that I did with scientists at the University of Utah some years ago. And what it's demonstrating there with fluorescence spectroscopy is that there's individual differences in bill fluorescence, which I also found with reflectance spectroscopy. One of the unusual things that I wasn't expecting was that in 2016, 11% of the breeding adult crested auklets had incomplete acquisition of bill pigmentation, like what you see in this slide. What you see here is characteristic of a phenology of late April, early May, when the birds are completing pigment acquisition. But this was summer at Diomede when the picture was taken. None of the birds exhibited this characteristic in 2015, and I'd not observed it in previous years or at other colonies. So there's something different about 2016. Now, auklets nest in rock crevices under talus slopes. They occur in very large breeding colonies that are synchronized in their nesting behavior. At Diomede, these birds need to have established nest sites by latter June. But in 2016, the attendance of crested auklets was reduced by 35 to 50%. This picture is from the eastern side of the island. And this is a picture from the western side of Little Diomede. And so as you can see, most of the island is steep talus, and most of that talus or habitat is occupied by seabirds. At Diomede, there's approximately 1 million crested auklets nesting at the colony, and a huge number of these birds were missing in June of 2016. Now, during 2016, a marine heat wave intensified in the Bering Sea. There was a heat wave in the North Pacific during 2014 and 2015, but during the winter between 2015 and 2016, it seems that there was a transfer of heat across to the eastern Aleutian Islands that resulted in this additional accumulation of heat energy in the marine environment. And so what this is showing you a plot of differences in mean sea surface temperatures. This happens to be between July 1st and 31st. This is looking at the additional heat in 2016 compared to 2015. So this is probably the highest heat content that's been recorded at high latitudes, at least in the northern Bering Sea and adjacent Chukchi Sea. So it appears that probably what happened was that due to this heat wave, it disrupted the marine food web and the availability of prey. Now, this is a plot of stable isotope ratios from red blood cells of crested auklets. It compares 2015 versus 2016. What you see there in blue are the data points from 2015. And you'll notice that they're relatively focused, kind of tightly constrained. And that's typical of this specialist forager that typically feeds upon large zooplankton. What you see in red is the diets or consumption patterns as shown from 2016. And you can see it's a much less constrained picture here. We have 3 times the range, essentially. What I should mention is that the horizontal axis shows the ratios of heavy isotopes to light isotopes of carbon, and the y-axis shows the ratio of heavy nitrogen isotopes to light nitrogen isotopes. Now, just to remind you, stable isotope ecology is informed by ratios of heavy and light atoms, in this case, carbon and nitrogen. The distribution of these isotopes in tissues provides information about patterns of consumption. For example, as nitrogen is passed up through trophic levels of a food web, it fractionates in such a way that the proportion of the heavier isotope increases. So in 2016, the nitrogen stable isotope ratios were higher, indicating foraging at a higher trophic level. The carbon stable isotope ratios were lower in 2016, and I interpret this to mean that the crested auklets had less access to the advected food web that is enriched from ocean upwelling. Now this figure shows the absorbance spectroscopy for species of euphausids that crested auklets feed upon. And the thin dark tracing superimposed over it is the UV vis spectrum of a crested auklet bill sample from Diomede in 2015. Notice how the absorbance band of the bill sample overlaps and appears to include the absorbance band of the prey. Euphausids are fast swimmers and can be difficult to capture even with a research vessel and nets. So a bright bill in this case may be advertising foraging success. I fabricated crested auklet decoys to test a hypothesis about bill fluorescence. The decoys are identical except for bill color. The control has an orange bill, and the experimental has an orange bill that includes fluorescence. You can see in the lower figure here reflectance of the decoy's bill types measured with a spectrometer. And you can see there in the red that the fluorescent decoy has a high reflectance than the blue, which is the control. And then you see a crested auklet bill reflectance there in the orange. And so it's in the same wavelengths as they would typically see in terms of reflected light of their conspecifics. So I tested the hypotheses that crested auklets would not prefer decoys with fluorescent bills. I had these behavioral assays where pairs of decoys were spaced 1.5 meters apart. I had 6 decoys that I had randomly assigned between the different groups. And then I had crest feathers that are essentially from crested auklets that were taken by the subsistent hunters at Diomede, the native people there. So the decoys for all extents and purposes are identical except for the bill color. So then this is showing you a picture of a crested auklet approaching a decoy. Just for simplicity's sake, I'm only going to discuss data with regards to approaches here. An approach was classified as within one body length of the model. And so what I observed, and this is showing you the approaches to fluorescent there in the red triangles, and then approaches to the control there in the blue circle. And then observation in number of hours across the bottom. Use a Wilcoxon mesh pairs signed rank test and showed that crested auklets approach models with fluorescent bills at a higher frequency. I had hypothesized that the pigment could have a physiological role as an antioxidant, as has been shown for other pigments. So antioxidants are important for mediating oxidative stress. T-bars is an assay that measures concentrations of malondialdehyde in plasma, according to the scheme shown in this figure. Malondialdehyde is one of the final products of polyunsaturated fatty acid peroxidation in cells. An increase in free radicals results in an increase in production of MDA. So MDA is a biomarker of oxidative stress. I use a colorimetric assay in a plate reader to estimate MDA concentrations. So I tested for a relationship between the pigment in plasma, and I inferred relative concentrations of the pigment in plasma with optical density at 245 nanometers. And use a quartz microplate because standard microplates have too much background noise, essentially, to get a good signal. So you have to use a quartz plate. But 245 nanometers is what I hypothesized to be the lambda maxima for the pigment based on what I had observed in extracts of the bill pigment. And then compared that to malondialdehyde as measured with the T-bars assay. And so what I found was that MDA was negatively correlated with the relative pigment concentration as measured by that method of optical density at 248 nanometers. So it suggests then a relationship between concentrations of the pigment in plasma and protection against oxidative stress. So in conclusion, bill fluorescence is a preferred trait. Crested auklets approached models with fluorescent bills at a higher frequency. The fluorophore may function as an antioxidant, as shown by the T-bars assay and the inferred relative concentrations of the pigment in plasma. Bill fluorescence and euphausid prey share chemical properties as illustrated by UV vis absorbance. expression of the bill fluorescence appears to be affected by ocean ecology, in that in the year of a marine heat wave, there was incomplete pigmentation, and that was also related to higher variance in consumption patterns. So in conclusion then, behavior, diet, and ocean ecology are linked with bill fluorescence in crested auklets. This research was funded in part by a grant to the University of Alaska from the National Institutes of Health. I'd like to acknowledge the native village of Diomede, which permitted me to do the work, Diomede School, Erickson Helicopters, campuses of the University of Alaska, Cincinnati Zoo, and others. So thanks for your attention. Thanks for the opportunity to present. And if you have any questions, I'm happy to try to address those questions.
All right, thank you so much for the talk. If you have any questions, you can drop them in the chat or in the Slack, and I'll read them out. I guess maybe while people type, I can start off with a quick question. I guess what are, I had one, it just escaped me. I guess you mentioned that in the years of the heat wave, there was high variability between individuals. And so I guess is there some, and you mentioned that they tend to come close to individuals with the more fluorescent beaks. Are these approaches to the highly fluorescent beaks also kind of an idea to measure sexual selection and preference in who you're going to mate with? And so I guess if you have these heat waves, does that influence the population dynamics of who gets to mate or not during those years of heat waves?
Well, it certainly could. I think the inability to express the pigment, suggests that you don't have enough of it. And it could be that the prey resource is limited to you. In other words, perhaps you can't get the U facets. And so if you do, now keep in mind, the birds may have a more sensitive vision for this than we do. Some of us looking at these bills might not pick up with the type of discrimination that the birds can. There are differences, for example, between subadults and adults. The subadults' bill color is relatively dull compared to the adults. And you can, when you with the instrumentation, you can see a difference in both reflectance and with respect to fluorescence. And the more intense the color, the more reliable the fluorescence signal. So I suspect that they can, and it makes a difference with respect to who you may select for mating. Now, one other thing that I would add that I didn't include in the presentation is that the crest, which is a basis for mutual sexual selection, to the extent that I've studied it, there's a correlation between crest size and the highest fluorescence that we measured, but that's sort of like a separate topic. But that suggests that there could be sexual selection there. But at this point, don't really know. Just the fact, though, that in terms of the marine heat wave, just the fact that you have birds not showing up on time to breed, that there is like leaving a hole essentially in the population for that year. Because if you're not there in time to breed, in the Arctic, you're not there. You're not there to breed. You're not going to be successful. Now, some of the birds did appear to show up later, but by the time they arrived, there was no chance that they were going to be successful in breeding. It's highly synchronized in terms of when birds breed. I hope that answers your question, but feel free to ask more if you want.
Yeah, so there's a few other questions. Let's see if we can get through some of them. One is there any interest in identifying the pigment and demonstrating its uptake from the prey?
Yeah, that's what I've been trying to do. Now, in terms of identifying the pigment, I can tell you lots of things that it's not, but I haven't been able to actually identify what the pigment is. I mean, I can say, for example, it's not carotenoids. And I have a hunch as to what it is in terms of the class of compounds, but trying to get it nailed down. So far, everything that I've tried to compare it to, it's not. It's similar, but it's not. So it could be a novel compound. Now, to actually identify, it will probably require, NMR as well as liquid chromatography and mass spectroscopy or mass spectrometry, excuse me. But the challenge in general with that kind of thing is if you can have the molecule be stable for you when you get it isolated. And so that's one of the things I'm working on. Then in terms of how to determine whether it's actually the prey that's the resource, that's something that I've attempted to do some work on. I've already tried to do NMR, and I've already tried to do LCMS. And the problem was not getting a pure isolated molecule. So there's work that I have to do yet to figure out how I can isolate the molecule by itself. And then when I do isolate it, hopefully it will remain stable. Right. Sometimes they don't.
Sounds good. There are a couple other questions in the chat, but I think we need to move on to the next talk. So maybe you can answer them either in Slack or in the chat later.
I'd be happy to answer things in Slack, and thanks so much.
Thank you.
Awesome, thank you. So our next talk is by Anke Klug, and who will be talking about effects of sex and mating in the presence and absence of a protective microbe. Getting that one?
Wait, I think I need to swap my screens, right?
Oh, I see it. That looks good.
You see the presenter screen or the
presenter view? Yeah.
Okay, wait, I just quickly switch it. Perfect. There we go. Now it should be better.
Yeah.
Okay. So welcome everybody, and I will talk to you about the second chapter of my PhD thesis. So I will talk about sex matters, the effects of sex and mating in the presence and the absence of the protective microbes. And I just wanted to quickly acknowledge my co-authors. So I had a wonderful master student who helped me with this project. I worked together with a postdoc who helped me set up this project and develop it further, and then my PhD supervisor. So I guess when I say that males and females are different, I don't really need to explain it that much in detail in this group of people, because we already heard a lot of examples today of how the two sexes are behaving differently, or are in fact showing differences. So when I say that females have barely any intrasexual competition, so females don't really fight among each other over mates or something comparatively, males have a very costly intrasexual competition. So normally males are fighting over being able to mate with a female, and these fights can, in the worst case scenario, even lead to death. On the other hand, females have a very costly production of the egg, while males have a rather cheap production of their sperm. Due to these two arguments that are listed here, it doesn't come as a surprise that females are rather having fewer offsprings, but with higher quality. So if you plot this as the number of offspring in terms of reproductive success, females optimize their reproductive success by having the optimal number of offspring. While for males, they actually optimize, or rather maximize, their reproductive success by a higher quantity. So for males, it's better to have the most offspring, while for females, it's better to have an optimal number. This principle, what I'm describing here, is known as Bateman's principle, where males and females invest differently in their offspring. But to be able to fulfill this Bateman's principle, each organism only has a certain amount of energy available that each organism needs to assign to either reproduction or immunity. And one could imagine that one is assigning these on the same basis, so that you invest the same energy into reproduction and immunity. But actually, there's discussion that females invest a lot more into immunity to be able to be healthy enough to then produce a high quality of offspring. While for males, it's hypothesized that they actually invest a lot more into reproduction to be able to contribute their sperm to the female. And if they die straight after of an infection, then they've still done the evolutionary purpose. So what we thought about is how would this energy investment change if you would bring protective mutualism into the play? So protective mutualism is defined as microbes that supplement the host's immune system. And it's known from a variety of organisms, not necessarily model organisms, but it's described in the cacao tree, in the Drosophila, and also in the North American bullfrog. And in my most favorite organism, in C. elegans. So if you now have already some energy invested into immunity because you invest energy into maintaining the protective microbe, do the two sexes actually invest their energy resources differently? And do we see a change in the phenotype? So we hypothesized that we would see increased reproduction in the presence of the protective microbe for females, while we hypothesized that we would see an increased survival with the protective microbe. And to answer these questions, or to work on these hypotheses, I have chosen my favorite model organism, C. elegans. So normally C. elegans is known as a self-fertilizing hermaphrodite, where within one organism, sperm and egg are produced and are self-fertilized and then just being popped out as fertilized eggs. Well, here I was using hermaphrodites that carried a specific mutation that enabled them disabled them to produce sperm, sorry. So basically these hermaphrodites, I refer to them as females because they do not have sperm, but they only have eggs. To nevertheless be able to actually have like fertilized eggs, there are males present in the population. You can find these males naturally in C. elegans populations, but at a very, very low ratio. Here, however, we have a very stable sex ratio of 50-50. To now test for costs of sex and mating, I set up a mating grid, which I will quickly walk you through so that you understand what I've actually been doing. So C. elegans has 4 different mating statuses. So in the L1 to L4 stage, it's impossible to distinguish between the two sexes. So the male and female will look the same. So I raised them in mixed population until I was actually able to say which ones were males and females. Once I could distinguish between males and females, I picked them into females only, into males only, or into 50-50 sex ratio plates. I left these worms there for 6 to 8 hours to allow them for mating. And once I could observe eggs, I would take that as a positive confirmation that mating had occurred because I could observe eggs. And after that, I then picked these worms once they were adults, I picked them into either not pre-mated individuals for males and females, pre-mated or pre-mated individuals. And due to this, I have 3 different treatments, which is for the female and for the male. I have unmated females, where I have these females that have only ever seen other females once they were sexually active. Then I have these short-term mated females that have been with males for these 6 to 8 hours, but were afterwards being picked on plates where they were again females only. And then I have 4 different setups, so the fully fledged setup for the different combinations of not pre-mated and pre-mated males and females. And the same is true for males. And here it's important to note that on these 2 plates and on these 2 plates, there's only one sex. So here on these plates, I have only females, and on these plates, I have only males. And if you recall my title, I talked about protective microbes. So I also quickly would like to introduce the bacterial diets that I was using. So I was using Salmonella, Enterococcus faecalis, and Staphylococcus aureus. These are now a lot of different species names that I'm throwing at you. So to make this talk a bit more understandable, I will refer to Salmonella as food, to Enterococcus faecalis as the protective microbe, and to Staphylococcus aureus as the pathogen. Why I do that, I will explain to you now. So in C. elegans, you can very easily do something called survival assays, where you infect the worms and you come back after a certain time, and then you count how much of the worms that are actually present on the plate are dead and how many are alive. So when I do this, I would, when I raise worms on food alone, and then once they reach the L4 stage, so just before they get adults, I infect them with a pathogen. I come back after 24 hours and I can see this amount of survival here. Once I now grow worms on the protective microbe and food alone, so I always need to mix the protective microbe with food because the protective microbe alone is not able to support full worm development, and transfer them to food later on, I can see that the protective microbe is barely causing any dead worms. If I now, however, have worms on the protective microbe first and then transfer them to the pathogen, I can see that there's a lot less worms dying, as this survival index is higher than the one on the pathogen alone. And this is why I refer to Enterococcus faecalis as the protective microbe, because it protects worms from infection with the pathogen. In this project, I had 3 different food setups. one, which is where I only ever grew worms on food, on the food bacteria. So I only ever transferred them from one food plate to the next food plate so that I could distinguish between adults and their offspring. And then I have worms that I grew on food, and then I transferred them to the pathogen. And then I have those worms that I grew on the protective microbe and food, and then later on transferred them to the pathogen. Just to keep it simple, I will refer to this as food, pathogen, and the protective microbe and pathogens. So with this setup that I just explained to you, I wanted to answer the question, are there differences between the sexes with microbe-mediated protection? And for this, I was investigating pathogen infection, the lifespan after worms had been infected with a pathogen, differences in behavior between males and females, and also depending on the different mating statuses, and whether I would see a difference in offspring production. Starting with pathogen infection, I again measured the proportion of survival as the graph that I showed you before. So the higher it is, the better. So 1 means all of the worms that are present on the plate are still alive. And on the x-axis, I plotted the sex and mating status, which is the same for almost all of my graphs. So you can see the females always on the left and the males always on the right. And then down here, there's the unmated, the short-term mated, and the lifetime mated. And the darker the color, the longer the worms have mated. So when I check for survival on the pathogen alone treatment, I can see, and I guess you will all agree, that lifetime mated females are doing a lot worse than unmated or short-term mated females. When I now check for proportion of alive worms in the males, I can see that these males are very high and there's no effect of mating whatsoever. If I now look at this in the presence of the protective microbe, I can see a similar pattern, as I can see that males are not affected by mating. However, I now see 3 distinct groups of females. So I can see that unmated are significantly different from the short-term mated and the lifetime mated ones. So summarizing this pathogen infection, I can say females suffer more during pathogen infection, which is mainly driven by the long-term mated females, and that males are not affected by mating. Once I had these worms infected with the pathogen, afterwards I would remove them from the pathogen plate and I would check for their lifespan. So I would transfer them to new plates every 24 hours to be able to track the exact adults and then see how long they were living for. So for this, I have plotted the proportion of alive worms here on the x-axis, on the y-axis, and I've plotted the lifetime in days on the x-axis. The different colors show the different treatments, and the different lines that will appear show the different sexes. So here, the female will always be on the left, and the male will always be on the right. So I have these 3 different diets for food, pathogen alone, and protective microbe and pathogen. And when I checked for food, which is kind of my baseline, I could very obviously observe that lifetime mated females are actually doing a lot worse than their counterpart females. While for males, I do not see a difference. And interestingly, this was the same pattern across the diets. The only thing that varies here is how long worms were living. So while you can see here, worms were living up to 25 days, here it's only 15, and here's maybe 15 to 20. So I can summarize that lifetime mated females survive the worst, and males are yet again not affected by mating. And while I was performing this pathogen infection and lifespan experiments, I observed a kind of different kind of behavior. So here, this picture shows you an agar plate, and in the middle, so this dark yellow spot is the pathogen spot that I spotted there before I added the worms. And all of these lines that make this plate look very pretty are in fact the lines where the worms crawled through and the bacteria then started to grow on the agar. So some plates would look like this, while others were almost clear. So this is obviously like a very hard trait to measure. So I tried to put numbers on this. And what I did for this is that I plotted the proportion of worms at the edge. So basically, whenever you look at an agar plate, there will always be some worms that you can't see because they're so close to the edge of the plastic dish that you need to slightly like flip the plate. So I plotted these worms that I would find there as a proportion of all the worms that were present on the plate. And whenever I did that for those worms that have been infected with the pathogen alone, I could observe that those sexes that had the other sex present on the plate were to be found at the edge at a way higher proportion. When I now checked for the proportion of worms at the edge in the presence of the protective microbe, I could observe a similar pattern despite it not being that clear as it was before. When I was doing this, I also realized that I had a high proportion of missing worms. So I always knew how many worms I had actually exposed and infected at the beginning. And very often I would not find a lot of them. So I was wondering where they went, and I was wondering whether there would be any link between some kind of mating and or sex treatment. So what I plotted here is the proportion of missing worms, which I could calculate as I had the originally exposed worms minus the live ones minus the dead ones. And the number that's left over is the worms that are actually missing. So these ones are plotted here. And here I can only see that males are going missing with a higher proportion than females are. And I could observe a similar pattern when in the presence of the protective microbe, as I can see that males are going missing with a higher proportion than females. And I can even see here that there's a significant difference between those females that were lifetime mated versus those females that were unmated. I furthermore wanted to check whether this proportion of missing worms would be the same over lifetime. So what I plotted here is the proportion of missing worms at the end point of each experiment, because it plateaued after some time. So if you want to see how it changes over time, I can just show you the graph, but it's a bit messy. So I decided to show this where I only plot the last time point of the experiment. And very interestingly, on food, I can observe what we already saw before, that lifetime mated females go missing with a higher proportion than unmated females, while it's actually the opposite for males. So for once, you can see, and I guess everyone agrees, that males are going missing with a lot higher proportion than females do. So this is 60%. So in some groups, it's like half of the worms that are actually picked on the plates are going missing across the evolution, or across the experiment. So this was a very interesting pattern. And when I then checked for on the pathogen alone, I could not observe the same pattern. What is, however, interesting to observe is that this y-axis is at 12.5%, while this one is at 60%. So there's a way lower proportion of missing worms generally. And this proportion of missing worms then increased again to around 20% when I would have the protective microbe present. And here I can again observe the pattern that males are going missing with a higher proportion than females do. So summarizing these two behavior results that I just showed you, I can say that males show a higher proportion of missing worms, and that I can see the highest activity on the plate when I have both sexes, so males and females present on the plate. And last but not least, I wanted to check for offspring production, because I realized that on some plates where I had mixed populations, I could not observe any worms. So what I compared was that I had pre-mated females, and not pre-mated females. So these females that were together with the males for 6 to 8 hours versus those females that were not. And whenever they are in this mating treatment, they were then directly already infected with the pathogen. So when I checked for pathogen alone, and then I compared not pre-mated females versus pre-mated females, I counted the proportion of plates that had offspring. I wanted to count offspring, but unfortunately that was not feasible. So I had to decide, I had to score the plates, either they had offspring or they had no offspring, so like a black and white measurement. And what I can observe, even though it was only a black and white measurement, was something very striking in observing that only 3/4 of the plates showed offspring, while when the females were not pre-mated, while those females that were pre-mated were showing offspring on almost all of the plates. And in comparison to this, I checked for the same amount of offspring present in the presence of the protective microbe, and I could not observe the same pattern. I was then also interested to see whether this is actually can be rescued over lifetime, or whether this maintains or whether this is rescued. So here I checked for food, and I could not observe the same pattern, neither in the absence of the protective microbe or in the presence of the protective microbe. So summarizing this, I can say that I have reduced mating without the protective microbe, and that this can be rescued over a lifetime. So now I have shown you all of these results, and in fact, it looks quite messy and it's a lot of results. So how do I actually make sense of this? So if I compare males and females, I could see that females have are able to invest more into egg production once they are in the presence of the protective microbe, while males are actually showing a higher activity, which is indicating that there might be higher mate searching behavior. So coming back to my original hypothesis, where I was hypothesizing that females would show increased reproduction while males would show increased survival, this hypothesis was only partially true because I can observe that both sexes have increased reproduction in the presence of the protective microbe, despite it being different as egg production for females and higher mate searching behavior for males. And with this, I would like to thank you for your attention, the organizers for setting up this amazing conference. It's in the middle of the night in Germany, so I would never be able to attend otherwise. But with this, I could actually show and share my research with you. And I would also like to thank my co-authors as well as my funding buddies and my supervisor. And I'm happy to take any questions. Thank you.
Thanks for the lovely talk and the beautiful experimental design and all those results. I think we ran out of time, but maybe just one quick question that popped up in Slack as well as where are the missing worms going?
So that's actually, so some of the worms, so if you put the agar in the plastic dish, then there's always like a tiny gap where the worms crawl into. So if you're unlucky, then they just crawl into and they crawl out again. But because I transfer them every 24 hours, they cannot come back. And sometimes they also make the very poor life choice of just crawling up the plastic edge, and then they just dry out there, so they just die. So that you just have to exclude them because you don't know whether they died due to the pathogen infection or due to the not having enough moisture, basically.
Mm-hmm. All right. I think we're off to the next one.
Yep. Okay. So our last talk today is by Madeline Gamble, who will be telling us about the role of intralocus sexual conflict in maintaining alternative reproductive tactics.
Thank you. Can you all hear me and see my slides? Awesome. Thank you so much for the nods. So I'll start by just thanking you all for making it this far with me. We are rapidly approaching what is my bedtime on the East Coast, but I'm really excited that you all have stuck around to listen to me tell you a little bit about my research. So I want to note that this work was inspired by my work on alternative male tactics in salmon, but the model I'm going to present here is not meant to be specific to anyone's system. So our current understanding of the maintenance of alternative reproductive tactics includes a few key mechanisms. And these are frequency-dependent selection, which I sort of think is everyone's favorite, disruptive selection, and female preference for novelty, which we heard a bit about earlier tonight. And these are all variable forms of selection, which we know is important for maintaining variation in nature. But importantly, these all focus on selection acting directly on the sex that is expressing the polymorphism, in most cases males. But that same genome is also experiencing selection in the other sex, in most cases in females. And so this led me to think about another form of variable selection, which is sexually antagonistic selection and intralocus sexual conflict. So we already know that males and females are often subject to antagonistic selection, and that this genomic tug of war can lead to the maintenance of variation. So inspired by the concept that the same trait can be advantageous in one sex but detrimental in the other, I wondered whether a three-way genomic tug of war could play a role in maintaining sex-specific polymorphisms. In other words, if genes determining male alternative reproductive tactics differentially affect the fitness of their daughters, could those male tactics be eliminated or maintained in the population by selection against or for their daughters who carry their genes, even in the absence of direct selection on the males themselves? And I want to point out that there is a growing body of evidence for genetic correlations between male tactics and female fitness, and that this occurs in both vertebrates and vertebrates. Sorry, I'm not sure if I said vertebrates and vertebrates, but I meant vertebrates and invertebrates. So I wanted to address this question using a model. So I built an individual-based model in which each individual's phenotype consisted of its sex and its body size. And body size was a threshold liability trait such that males that have a body size above the threshold develop into alpha males, and those with a body size lower than the threshold develop into beta males. And I also set up the model such that female fecundity was correlated with body size, such that larger females had higher fecundity. And importantly, this sets up the opportunity for intralocus sexual conflict if selection favors large females but small males, and body size is heritable between the sexes. So I built this model, I ran it for 500 generations each time, and I'll tell you a little bit more about it. I made some critical assumptions. One was that the phenotype is entirely genetically controlled. Another is that all alleles are inherited in Mendelian fashion. And finally, there was no evolution of the threshold allowed in this model. And these are all assumptions that I would really like to explore relaxing later, but for tonight's presentation, they hold. And finally, this model was genetically explicit, and I'll go into that a little bit more now. So in my model, each individual had 20 loci. And at each locus, an individual had 2 alleles. These were diploid individuals. And the alleles are here represented by these black boxes. And each allele had 2 possible values that it might express depending on the individual's sex. And those are represented by the blue and the red boxes here. So this allowed me to alter the way in which the body size phenotype was calculated in different model scenarios. So for example, when all loci were general in their expression, the 2 values within each allele were averaged, and that averaged value contributed additively to the phenotype. When a locus became sex-specific in its expression, only the male or female value of each of its alleles was expressed and contributed to the phenotype. And this is the biological equivalent of sex hormones altering gene expression. Finally, when all loci are made to be sex-specific, I was able to get the evolution of complete sexual size dimorphism and effectively an intersexual genetic correlation of 0 for body size. So we'll come back to that in a moment. And with this in mind, I'll return to my main question, which was under what conditions can intralocus sexual conflict maintain alternative reproductive tactics? So I'll walk you through these different conditions that I modeled. First, I modeled a variety of selection regimes on male body size. The first was a selection regime in which male body size was not selected on at all. The second was a negative frequency dependent selection regime in which the rare male had the fitness advantage. And I started off with 3 different starting ratios to make sure that the initial frequencies of males didn't affect the outcome. So these were a 3-to-1 ratio, a 1-to-1 ratio, and a 1-to-3 ratio. And finally, I also modeled a directional selection regime in which beta males were favored. And I also had 3 different subsets of this selection regime in which selection favoring beta males was weak, moderate, or strong. And then finally, and going back to thinking about the model being genetically explicit, I modeled different degrees of intersexual heritability of body size. So when intersexual heritability is high, sons closely resemble their mothers and daughters closely resemble their fathers. But as the number of sex-specific loci increases, intersexual heritability should decline, such that sons no longer resemble their mothers and daughters no longer resemble their fathers. So this combination of 7 different selection regimes and 5 different numbers of sex-specific loci resulted in 35 total modeled scenarios. And I had 2 predictions about the conditions in which alternative male tactics might be maintained. The first was in the modeled scenario where there was no selection on male body size, but low intersexual heritability of body size. And so that means that larger females do not necessarily produce larger sons. The second, and this is our intralocus sexual conflict scenario, is when there's directional selection favoring beta males, but high intersexual heritability of body size, such that alpha males, though having lower direct fitness, produce fitter daughters. And I'll just note here that as we go through the results slide, I'll call your attention to the results in which intralocus sexual conflict is acting with this little red starburst. So I want to first give a summary of the results because I'm about to show you quite a number of figures. The first thing we found was that increasing the number of sex-specific loci decreases the intersexual heritability of body size and facilitates the evolution of sexual size dimorphism. And these were reassuring results to see. Second, we found that sex-specific selection gradients were affected by the number of sex-specific loci and the selection regime. And again, that was a good check to see that our model was producing the kind of results we would expect it to produce. We also found that gender load is maintained when the sexes cannot evolve independently, and that, consistent with theory, population size is limited when the gender load is maintained. And finally, we found that alternative reproductive tactics can be maintained by two mechanisms other than negative frequency dependent selection, one of which includes intralocus sexual conflict. Okay, so here's the first of those results. On the x-axis is the number of sex-specific loci, and on the y-axis is heritability. And here you can see that the more sex-specific loci we had, the lower intersexual heritability became. So in these box plots, the darker colors are sire-son and dam-daughter heritabilities, and the lighter boxes are opposite sex heritabilities, so sire-daughter and dam and son. And this particular figure comes from a no selection regime, but the others look qualitatively the same. Second, we found that sex-specific loci facilitate the evolution of sexual size dimorphism. So here, again, we have the number of sex-specific loci on the x-axis, the mean body size of both males in white and females in gray at the end of the 500 generation simulations. And I have these plots up here for the no selection regime, the negative frequency dependent selection regime, and the directional selection against alpha males regime. And you can see that in each scenario, sexual size dimorphism did evolve, but importantly, when there were no sex-specific loci, sexual dimorphism was not able to evolve. Okay, so the next few graphs are a little complicated, so I wanted to just walk you through them first. Again, we have the no selection regime, the negative frequency dependent selection regime, and the directional selection regime. On the x-axis, we now have generation, so from 0 to 500. And on the y-axis, our variable of interest in this first case, it's the selection gradient. So first, I'll show you, we found that sex-specific selection differs among selection regimes and the number of sex-specific loci. So the final tricky piece about this slide and the ones that follow is that each one of these figures now has a separate box for the number of sex-specific loci from 0 on up to 20. So you can see first that the sex-specific selection gradients that we calculated from our models reflected the imposed selection regimes for at least the first few generations. So you can see in all cases that selection gradients on body size in females was positive initially, and that in the directional selection regimes in which beta males were favored, selection on male body size was negative initially. Now, in most scenarios, you can also see that these selection gradients for both sexes converged on 0 as the populations responded to selection and body size evolved. But importantly, a few scenarios resulted in differences in sex-specific selection gradients. And this was the negative frequency dependent selection scenario and the directional selection scenario when there were 0 sex-specific loci. And this is a consequence of high intersexual heritability, which constrains the evolution of sexual dimorphism and maintains the gender load. So again, in concordance with theory on sexual dimorphism and sexual conflict, we can see that when antagonistic selection is maintained, population growth rates are dampened. And this is, again, reflective of the gender load. And finally, as we predicted, alternative tactics were maintained in 2 scenarios besides frequency-dependent selection. So in this case, I'm showing you the frequency of alpha males in black and beta males in gray across the 500 generations that I modeled. And the first of these scenarios in which both tactics were maintained is our intralocus sexual conflict scenario. So in this situation, we have selection acting against alpha males or against large males, coupled with high intersexual heritability. And this meant that alpha males were maintained via selection for their genes in females. And importantly, I'm not showing it here, but this figure of directional selection is from the moderate strength of selection. And this maintenance of our alternative tactics is only possible when the strength of this directional selection sort of equally balances the strength of fecundity selection on females pulling body size to be larger. So the second scenario in which alternative tactics were maintained was the no selection regime coupled with low intersexual heritability. So in this case, females are selected to be large and are able to attain that large body size themselves, but cannot pass that on to their sons. And finally, an interesting outcome that we did not set out to address is that we found that male tactic can go to fixation even when males do not experience direct selection. And this occurs through correlated selection on females. So I'll remind you that in this no selection regime, male body size was not under any kind of explicit selection. However, in these lower numbers of sex-specific loci, males were basically pulled to a larger body size through correlated fecundity selection from females. And this further highlights the importance of considering selection in both sexes when we're understanding sex-specific polymorphisms. So to conclude, existing mechanisms for alternative reproductive tactic maintenance focus mainly on selection acting directly on the sex exhibiting the polymorphism. But if we expand our focus to include both sexes, we find two other mechanisms besides negative frequency dependent selection that are able to maintain alternative tactics over at least 500 generations. And these include intralocus sexual conflict and the situation in which there's no selection on males, but intersexual heritability is low. And finally, I just want to point out that this concept has really important implications beyond the evolution of body size or sex-specific polymorphisms and sort of into the realm of population ecology, in that we saw that with the maintenance of the gender load, population growth rates were severely limited. And so if alternative tactics are maintained through intralocus sexual conflict as modeled here, then their maintenance may have a negative effect on their population's demographics. So with that, I'll say thank you. I want to particularly thank David Noakes, who recently passed away, but was the first person who pushed me to address this question through a modeling approach. And it's something I've never done before, but I am happy I did, and it's because of him. And if you have questions, I'm happy to take them now or later via Slack, Twitter, or email.
Thank you for the fantastic presentation and for going through the last talk of today. Or I guess there's one more talk in an hour, but last talk of the session. There's one question that came in the chat. How could we search in real organisms for empirical signatures that would specifically support your theoretical predictions?
Yeah, that's a really good question. So this is the time of the day when my quick response brain has been off for hours, but there are a lot of things that come out of this model that are that I think can be used as those hallmarks. One of them is the maintenance of the gender load in what's the word I'm looking for? In conjunction, there we go, with alternative reproductive tactics. I'll think a bit more about that and follow up later, because I think that's a really good question. And that's sort of where I'd like to take this next is, does this actually happen? Just because it can happen doesn't mean it does, right? But figuring out where we might want to start out looking is a cool prospect.
Thank you. If anyone has any more questions, this is a good time to put it either in the chat or in the Slack.
I can ask a quick one. So are there meta-analyses of cross-sex genetic correlations that might tell you whether, which of those scenarios that you had is likely?
So there will be, once I write my third dissertation chapter, because I haven't quite started that process yet, the answer is I don't know. But I think that if there are certainly none that I know of that then connect those cross-sex genetic correlations with any sort of sex-specific polymorphism. And so I think that that's sort of the path that I'd like to take that question.
Thanks. I guess with that, we can thank all the speakers of today's session, the social behavior session. Thanks. Thanks Julia for co-moderating and answering questions, getting questions from the crowd part. And yeah, enjoy the rest of the talks. There's the presidential presentation is in an hour. So there's time to eat dinner for those who haven't done that yet. And then another day tomorrow.
Thanks, everyone. Thanks for staying.